making it political boundaries, but animals don't have any boundaries as such. So this is good for us for documentation statewide. So tomorrow, statewide, they can implement any actions or laws, whatever they want. So that's the reason the entire motion is about like to restrict to only Karnataka. So I'm going to talk about the amphibian discoveries that has happened in Karnataka and what is the past, present, and what is the future with respect to discovery. So if you see yesterday already, I guess Dr. Dinesh, one of the leading taxonomists in amphibia, would have explained you about what are amphibians, where they are found, and the global scenario and Indian scenario. So if I could start, I will not do all those things because it will be a repeat show. If you start with the discoveries, why there are so many species discoveries? What is the reason? If you see, there are a lot of mega diverse countries, and India is one of the mega diverse country, and it holds ninth place, being a mega diverse country. So what do you mean by this mega diverse? Means to say that having majority of Earth species and also highly endemic, or having high endemic numbers. In that, being a mega diverse, India stands 9 and Brazil being the first. And if you see that apart from being a mega diverse country, India, it also has the biodiversity hotspot. Of 36, India is also a country which is part of the biodiversity hotspot. Now, what is this biodiversity hotspot? It's nothing but a biogeographic region with significant levels of biodiversity that is threatened by human habitation. And these are the places where it's filled with species, diversity, and richness. And that's the reason there are a lot of discoveries happening across the globe, where you can see this map which shows all the places which are than when it comes to India alone. So if you see the biogeography of India, it's very unique compared to any other country in the world because India's biogeography is like it's different, it's diverse. You can see there are 10 different biogeographic zones in India, right from Trans Himalayas till the islands. So our country is something unique compared to the rest of the other countries across the globe because you see all these diversity is different like sorry the biogeography is different right from trans himalayas desert and the diversity of the animals also is different from each biogeography. When it comes to amphibia, you all know amphibians are cold-blooded animals and there are basically three classes. Your class amphibia, then your urodela and Echinid or the limbless amphibians. And when it comes to amphibian role in the ecosystem, they provide various ecosystem services. When you say ecosystem services, they also contribute something to the ecosystem. They are part of the uh, food chain. They also control the pest population, and they are being studied for various reasons. And they also help in sustaining a healthy ecosystem. They are very good ecological indicators. When it comes to Karnataka, because our study is mainly focused on Karnataka, why Karnataka is also special because Karnataka is being the seventh largest state in the country, and also if you see the physical map, the Karnataka map, the physical map, you can see again even it's diversified. You have the coastal zones, you have the hilly region, there's the Western Ghats, then you see the Deccan Plateau, which is divided into the northern dry zone and the southern dry zone. And if you see the Western Ghats, again you can see the majority of the forests, Western Ghats forest falls in Karnataka. And that's the reason you can see a lot of discoveries also are from the state. And if you see Karnataka, as I told you, these are the three different zones. You can see that you have the coastal, you have the hilly regions, and you have the Deccan Plateau into northern dry zone and southern dry zone. And even here, the diversity is different depending upon the ecosystem. When it comes to India, amphibian studies in India, it dates back to 1799, where the first 5 species were described. These are the 5 species which are described in the year 1799 by Snyder, and that's how it kick-started in India, the amphibian discoveries. So whatever you see, these are the first 5 species which were described in 1799 by Snyder. When it comes to amphibian checklist India, so Karnataka, in 2020, there are 102 species. And if you see the families, how they are arranged here, these are the families you can see Indo-Triplidae 4%, Bufonidae 7%, like that, all put together, a total of 102 species are found in Karnataka. Not discovered, but mostly reported from the state. Out of 102, 92 are frogs and toads, and the remaining 10 are the caecilians. When it comes to amphibian discoveries, we can see it dates back to 1876. The first species was described in 1876 from the Brahmagiri hills, known as Gatophrina ornata. So this is the first frog which was found in 1876, and the latest being the Tirutika Bengaluru, which was discovered from Bengaluru city. And when it comes to Sicilians, you have Digenus krishni, the year 1999 by Pillai. Then you have Ichthyophis davidi in 2011 by Bhatta. So these are the species which were found, but discovered from the year 1876 to present in 2020. So if you see amphibian discoveries in Karnataka, total of 55 species are being discovered from Karnataka. And if you see the checklist, it says 102, almost 50% have been described from the state. That makes the state also be proud that so much diverse species have been discovered from. Now, one more interesting thing, what I want to tell you is, see here, picture, all till now, whatever frogs have been discovered, most of the Western Ghats, like this, and next. And this say there are a lot of yet are awaiting the formal discovery. There are a lot of chances
I
anybody can.
believe Deepak, there's some audio issue. If you can check that.
So I don't. Am I audible now?
Yeah.
So I don't know where I went. So if you see, these are the species which have been described in the Karnataka. If you start from the beginning, the first amphibian species, the frog, was in the year 1876 by Gunther. That is Gatophrina ornata here. Then the latest one being Sperotheca bengaluru from the peri-urban zones of Bengaluru city. In case of Sicilians, the first one was Digenus krishni in 1999, and Ichthyophis davidi being the latest in 2011. So these are the discoveries from the past till the present. What about the future? If you see till now, whatever I told you, the species discovery, right? Till now, whatever the species discoveries which you can see here from 1876 to 2020. So most of them are, or almost everything is from the Western Ghats region of Karnataka and few from the coastal plains. Whereas the remaining are still unexplored. That is the Deccan Plateau, the northern and the southern dry zones. So this means to say that a lot of field explorations are needed in terms of sampling of the diversity, inherent diversity across these places. Probably we could encounter any new species. That's how we encountered this particular species from Bangalore. So that was part of the field work where we could do it. And there's a lot of opportunity for people. If anyone wants to work, they can explore these parts of Karnataka, and definitely there might be new species awaiting their formal discoveries. So that's my talk. Thank you. I acknowledge all the people who are part of this, like Dr. Dinesh, my guide Asha Devi. Thank you. Any doubts? Any questions?
Thank you, Deepak.
Thank you. Hope I was fast.
Yeah. Mr. Deepak?
Yes, sir.
Yeah, it was a wonderful presentation. In a very short span, you have given a clear idea about the statistics of the amphibians that were discovered earlier, as well those that were discovered in the recent past. You just showed Ichthyophis davidi. Yeah. When was that? In 2011, may I know who discovered that?
I told you, right, sir? It's Bhatta.
But okay, I couldn't hear that. All right. Wonderful presentation. Thank you very much.
Thank you, sir. Thank you.
Any questions from the audience, please? We just have time for one or two questions, quick questions. Any questions from the audience? You can raise your hands, unmute yourself and ask your question, or you can type it up in the chat box. no questions. Thank you, Deepak. We'll go on to the next oral presentation.
Sure.
This is from Lalpia Zula and Lalrem Sangha. Is one of you here? Lalpia Zula
Yes.
and Lalrem Sangha.
Yes. I can share my slide now.
Okay. So you're going on next. Kindly share your screen.
So, hello everyone. My name is Lalbir Zola. I'm a PhD scholar in the Department of Zoology, Mizoram University. Today, I will be talking on the topic of taxonomical insight on Cynomicrurus mcclellandy, population of Mizoram, Northeast India. First, let's have a look on the background. Coral snakes are venomous elapid snakes belonging to the 5 genera, which can be divided into 2 distinct clades: the New World coral snakes or the American coral snakes, which accommodates 2 genera, Micruroides, which is a monotypic genus, and Micrurus, accommodating 81 species, and the Old World coral snakes, or the Asian coral snakes, which comprised by 3 genera, Calliophis, Hemibungaras, and Cynomicrurus, and the Cynomicrurus comprising 8 species. And coral snakes are further grouped into 2 tribes under the subfamily Elepini, such as the Calliophini and Hemibungardi. So coming to the Indian scenario, currently there are 6 nominal species of coral snakes in India, Cynomicrurus mcclellandy, and 5 species of Calliophis. Regarding the focus species, Cynomicrurus mcclellandy, there are 4 distinct varieties of under this species, or some author can say use to mention it as subspecies. So the first one is variety typica. It is found in Mizoram, Arunachal Pradesh, and Assam in India. And the variety univirgatus, it is found in Sikkim, Nigriventar in Himachal Pradesh, and variety Gorei in Assam, Manipur, and Mizoram. So in Mizoram, the focus area, earlier it was first reported by Matthew 2007, and later Larem Sang and Zotan Siama provided 3 distinct morphological varieties of the species. Here we can see in the slide that the 3 types they are presented where here the type 1 is the one which having strongly banded forms, the one with black or small dots on the vertebral lines, and the one with the which are lacking dorsal bars or spots. And here are the challenges. Recent studies suggested that different color variants observed on the Indian subcontinent of this species and its vicinity are most likely represented by intraspecific variation, which need population-level molecular sampling to resolve this issue. Although Smart et al., with this recently published paper on the systematics of Cynomicrurus, they hypothesized the variants of Mizoram mentioned by earlier worker to be mutable chromatic patterns represented by intraspecific variation. However, we investigate in this study the taxonomical status of this mcclellandy population of Mizoram by adopting the combination of morphology and 3 mitochondrial genes. So here are the methodology which we followed in this study. Specimens were collected from different sites in Mizoram, and liver tissues were used for the genetic for generating the genetic data, and morphological examination were performed, and ventrals were counted according to the methods of Dowling, and DNA were isolated using the DNA mini kit, and PCR amplification were done, and sequencing were performed using Sanger's dideoxy method. And the table here, I am showing the PCR condition which I am using in this study. And then the generated sequences were concatenated, and the 3 genes were combined to form a single data set. And using that, Bayesian inference phylogeny was performed. And apart from that, two types of single locus-based species delimitation were performed. Here in the table, I've shown the models and the partitioning scheme used for the Bayesian analysis. Coming to the results and discussion, based on the recent paper that was recently published regarding this group of species, the variety gray differs from the other variants in the absence of black body bands and black dorsal stripes, and the dorsal stripes were replaced by small black spots and the black Nusselt band concave anteriorly. And in this study, we regard the specimen, the type 3 varieties mentioned by Narevsa and Zot and Siema as variety gray, and the total materials examined for this variety is 36 specimens, and for the variety typica, a total of 2 males and 1 female are examined. Here is a table showing the morphological comparative table, and here we can see that in the variety gray, the ventral is range from 208 to 241, while in the variety typica 195 to 218, and in the subcaudal 24 to 46 in gray and 27 to 31 in typica. And regarding the Nusselt band, although the Nusselt band was mentioned only as concave anteriorly, here we found that they are mostly showing a straight orientation, and some might have a mildly pointed edges at parietals anteriorly or even posteriorly. In the case of this variety typica, the Nusselt band, the black Nusselt band, is conspicuously narrowed at the mid portion of the band. So here the head portion of the 2 varieties. On the left side, we see that most of the Nusselt bands were in a straight orientation, but there are some specimen which pointing edges towards the frontal scales, and some very few specimens are having a concave towards the anterior side of the head. But in the variety typica, most of them, majority were having a conspicuous narrowing of the Nusselt bands from the mid portion of the Nusselt band. Going to the systematics, the final data set contains a total of 2,354 base pairs. Here we can see that the variety gray, which is shown in the top of the tree, it is forming a distinct lineage with respect to the variety typica here in a brownish, light brownish color. It is the variety typica. Here is the sample of Mizoram, and it is clearly distinct from this variety typica. And variety gray is showing within these 3 sequences of gray, we've obtained a genetic distance of 0.4 to 1.3%. And however, it was in the recent paper, it was recovered as a single species. In this study, we recovered a considerable genetic divergence among this clade. So going further, this Gorey variety is also showing a genetic distance of 10 to 10.5% with this nigriventer, and with the group of Painani, it is even showing 10.5 to 11.5%. So here is in the previous slide, I was only showing this portion of the top portion of the phylogenetic tree. Here is on the left side, the whole is shown here, and in the right side is the recently published paper which I'm talking, because it was recently published, and I have to mention a couple of times because they also addresses the species of interest which I am dealing with here. So here we can see that most of the clustering is accorded with this recently published species tree also. So the main challenging part is that in regard to this Taxonomic rules, McClellan clade, they are recovering as a distinct lineage, single lineage. But in our analysis, we recovered that there are distinct putative lineages in this Taxonomic rules clade, McClellan clade. But it is very difficult to make a conclusive suggestion on the status of Mizoram population so far, because in this study I am using only the 3 mitochondrial genes, but in the recently published work they're using nuclear and other mitochondrial genes like ND2. So in order to clarify or make a clear-cut statement, further research with more genetic data must be done. So apart from the systematics, additional distributional records are also presented in this study. Total of 7 new localities were presented for the variety typica, from which are represented by 4 districts, and regarding the variety Gore, a total of 14 new localities are presented in the studies. And the breeding habits are also observed in this study. One of our collected specimen from Durtlang was found gravid, and it was carefully monitored. And on 3rd September of this year, the female laid 3 eggs, and after exactly 1 month, the eggs were hatched, but unfortunately, one eggs didn't survive. During this incubation, the temperature range from the room temperature range from 24.7 to 31.1 degrees Celsius, and the relative humidity 70% to 19.3%. And the reported clutch size for the species is 6 to 14 eggs, but in this study it is quite less. Here it is we also speculated that there can be some slight difference in the clutch size of between the 2 varieties, because the egg size of the variety Gore is relatively large with compared to the body build of this female, and so that as far as we have examined, most of the Gore varieties are slender and smaller in compared to the variety typica, which are robust body build and a bit larger. So coming to the conclusion, the morphological feature of the 2 varieties of Mizoram population mostly agree with the descriptive characters of the variants provided by Smart et al. 2021. And the Mizoram population of variety Gore is recovered at a distinct lineage with considerable genetic divergence from the other putative lineages obtained in the BI phylogeny and genetic distance-based ASAP. It is assembled species by automatic partitioning. The taxonomy provided so far in Smart et al. Is provisional, and thereby suggesting further studies with more genetic data, particularly nuclear, for all the varieties, populations, and intermediate population to test their hypothesis. Given that Smart et al. Use a single specimen to represent the Mizoram population in their analysis, we suggest generating more genetic data, as previously mentioned, in order to fit the dataset of their analysis and test whether the color variance among Mizoram or even other Northeast Indian population of Taxonomic Rurus McClellandy is intra-specific variation, or the variety Gore is rather a distinct evolutionary lineage with definite certainty. Thank you.
Thank you, sir. Ganesh, sir, over to you.
Yes, ma'am. Thank you. Mr. Lal Bikazula, can you hear me?
Yes, sir.
Mr. Lal? Yeah, fine. It was a good presentation indeed. Your finding on the taxonomic insights of this Cynomicrurus. Generic name is Cynomicrurus, right?
Yes, sir.
Yeah, you said 6 coral
Yes, sir.
snakes are in India, right? Are
Yes,
all these 6
sir.
coral snakes of India poisonous?
Yes, sir. They are elipid snakes, so they are
What type of poison? What
known
type
to.
of poison? Neurotoxin or hemotoxic?
They
Are
are
all the
mainly
same?
neurotoxic.
All of them.
Yes.
Okay.
If I'm not sure, but I'm not wrong. Yes, sir.
Okay. Any mimics of this coral snakes that you have seen? Any mimics of the non-poisonous snakes that you have seen regarding these coral snakes?
Regarding the mimicry, in Mizoram, only I can say in the context of Mizoram.
Yeah.
So, yeah, I am not
Any
sure if we can
non-poisonous?
consider as
My
a
question is there any non-poisonous
yeah.
snakes that mimics these coral snakes?
Yeah, some oligodon snakes also mimics these coral snakes.
Okay.
They are also showing the the underbelly pattern of the coral snakes. Even
Okay.
their behavior, like the tail coiling.
Okay. Okay. In terms of look, coloration,
Yes, yes.
you have found some mimics.
No, in Mizoram, I didn't have any like a clear, very conclusive mimic to these coral snakes. But
Okay.
other part of the distribution there might be.
All right. Thank you very much.
Okay, sir.
Thank you, sir. Thank you. Any questions from the audience, please? Any questions? Okay, since there are no questions, we'll move on to the next presenter. Tessimson, are you here? Tessimson?
Think he's not there.
Oh, yes, I'm here, sir.
Okay, sorry.
Citation.
Fine. Our next presenter is Tessimson et al. So can you
Yes, sir.
kindly do your presentation?
Sure, ma'am.
You can share your screen.
Yes. Sure. So, ma'am, can I make use of Amit Kumar's BAL device?
Sure, sure.
Okay, thank you so much. Is it visible, ma'am?
Yes, sir, your presentation is visible. Can you just put it on the presentation mode?
So, is it audible, ma'am? My voice?
Sir, it's not too audible. Can you kindly minimize your Microsoft Teams screen also, sir?
Okay.
Yes, sir. You can put it on the presentation mode, sir.
Okay. Presentation mode. Okay.
Yes, sir. It's on the presentation mode. You can go ahead with your presentation.
All right, all right. Okay.
Thank you, sir.
So, hello, my name is H.T. Disempson, research scholar from Department of Zoology, Mizoram University, Aizawl. So, the title of my presentation is an updated checklist of amphibians in Dampa Tiger Reserve in Mizoram with a note on threats and conservation challenge. So the state of Mizoram is located in northeast India, and it's one of the largest. The Dampa Tiger Reserve, DTR in short, is one of the largest forest cover. It has the largest forest cover in the country, in the state, and is a home to some of the most cryptic and distinctive herpetofaunal diversity. DTR is the largest, has the largest area among the 10 protected areas in the state Mizoram. A biodiversity hotspot location, which is a part of the Indo-Burma hotspot, which serves a linkage to tropical forest in Southeast Asia, such as Bangladesh, Indonesia, and Myanmar. And so far, Pawar and Biran, for the first time in the state, and there were many new records from which Pawar and Biran has reported, of which 18 amphibian species. Then after 10 years later, myself and our team, we have reported an additional 10 more species. Now, based on those two reports, now we have updated another 7 more, of which there are 7 families and 24 genera. Now, the reason why we have selected these particular largest protected areas among the 10 is because the complex geographic settings are such that the topographic relief and the distributed habitat types and other insular systems, the endemicity, the environmental heterogeneity promotes differentiation. Then we also assess the local biodiversity, which would also greatly assist in decision making for environmental conservation and the management policies. Now, coming off from Dampa Tiger Reserve, in the northern part of the proper Aizawl, which is roughly around 80 kilometers from the city, there is a national wetland named Trumdine National Wetland. Some 10 years ago, Sen Gupta et al., including our professor H.T. Ramsanga, they have described a new species, a megophrid frog called Leptobrachila trumdine. And 10 years later, the same similar and identical species, Leptobrachila trumdine, was an additional record in Dampa Tiger Reserve. This was as per Vandal Siamoyi 2020. And furthermore, we have collected the same species, Leptobrachila trumdine, in the further south of the Dampa Tiger Reserve, including other sympatric species such as Rhocestus rezakhani and Silvirana lacrima, which was published beginning of this year. And it's an country record for these two, Rhocestus rezakhani and Silvirana lacrima from Dampa Tiger Reserve. Now, what our main highlight for this presentation would be the update additional amphibian diversity with input insights on the existing threats and conservation challenges of Dampa Tiger Reserve in Mizoram. Now, in order to conduct any survey, we need an official written permit, which we can obtain from the Chief Wildlife Warden in Aizawl. And the survey for this particular paper works, we have carried out a survey between February to November, which is roughly 10 months this year, conducted in both primary and secondary forest, along streams, ponds, rivers, any available from lowest to the highest gradient. Now, coming to morphology, we take the measurements on in the field itself, and which required any further description is collected. Samples were collected, and then they were morphometrics were noted by using caliper. Then they were preserved, and they extracted tissues for genetic analysis, and then they are tagged in the departmental museum. And then furthermore, for morphological genetic data and published literatures were also employed. Then gene sequencing data were deposited in the National Center for Biotechnology Information, India. Moving on, this is the result. These were the updated results from Dampa Tiger Reserve, which was already published in the beginning of the year. Now, this is the additional 7 new species based on our work. Now, the first one we have Raucestes manipurensis. As per the IUCN status, it's not assessed. Now, this would be the new record for the state. Second, we have Polypedes brori, which is a data deficient. Theloderma bibogens, data deficient. Then Curizales youngi, not assessed. Ichthyophis multicolor, data deficient. Buphoides megalayanus, which is an endangered. Now, regarding this Buphoides megalayanus and the last one, Buphoides kempi, it was mentioned that yesterday, point out by one of our speakers, that this is endemic to one of the northeast state, that is Meghalaya. That's now, this is including Mizoram, so it would be a record for the state as well. So Raucestes manipurensis and Buphoides, these two Buphoides megalayanus and Kempi species would be now further geographic distribution is widened. Now moving on to the species accounts, Raucestes manipurensis. The description of this particular species, the SVL, snout to length, snout vent length, ranges from minimum 17 to maximum 24 then millimeter, and the snout is obtusely pointed, slightly projecting, flanks yellowish with irregular brown patches. Now, as you can see in the screen, this is the location where we collected in Dampa Tiger Reserve for this Raucestes manipurensis. And below are the two different polymorphs, as it has been rightly pointed out by Lal Monsanga yesterday, speaker, that these are the two polymorphs, but a same species of Raucestes manipurensis. Now, moving on to that ecological nodes. 5 individuals of this species were encountered from small seasonal stream, and in the northern part of the Dampa Tiger Reserve near the anti-poaching camp, Thaichang Forest anti-poaching camp, and near the Tuipui Bari village road. Now, at an elevation of 495 meter in a semi-dried stream, and the habitat forms for this, the particular microhabitat for this are manipurensis were collected, were encountered near the shrubs, bamboos, and with the rocks. And other similar sympatric species found near R. manipurensis is Raucestes rezakhani. Second, we have the Polypedes brori, and it is characterized by a large head, as you can see in the slide. This species has got a comparative large head to that body, and has got a smooth dorsum with the numerous minute words. Then the ventral are mostly rough. Back of the hind limbs has several black bands, and this is as per Raman et al., which was described from Myanmar. Now, ecological nodes. We found this particular female species as shown in the slides from the banks of one of the streams called Tumkon stream on 24th of March this year. There are two ranges in Dampa Tiger Reserve. The northern part, they call it the Teire Range, and this is at the southern side of the Dampa Tiger Reserve, Puldungsei, at an elevation of 435. Usually they are mostly found, all these species, amphibian species, at the lower range. Now the surrounding habitat consisted of primary forests, mostly shrubs, grasses, and bamboos. The third, Teloderma bibugens. It has got a broad head, short snout, and the tympanum, a circular ring like just behind the eye, is quite distinct as it can be seen here. Then ventrally granulated speckle with dark brown, and the individual collected here is a male with a record morphometry of snout vent length 15 millimeter. It was collected from a soft mud near seasonal stream at Sial Ring, the exact locality. Then the surrounding habitat consisted of the primary forest composed of shrubs and thick canopy. And the surrounding ambience were suitable for amphibians at 19.4 degrees centigrade and a relative humidity of 80%. Now, Curis zealous youngi, this was described in the year 2018, and 3 years later, now we have found further down south in the Mizoram state from its holotype where it was collected. Now the description is it's greenish and brown colored with dark markings, distinct pointed snout. When viewed laterally, head longer and wide, tympanum is distinct, head is covered with small warts, body with coarse dorsal and lateral warts intermixed with small and irregular tubercles. And morphometry for this species is 35.79 mm. Ecological nodes collected from undisturbed riparian forest across different localities in Mizoram. This has been collected from the northernmost part of Mizoram, Pualreng Wildlife Reserve and Dampa Tiger Reserve. And the locality exact where we collected, it was found in the rock crevices. There is a hollow rock crevices near a fall called Kotlabung in Puldungsei range. The surrounding habitat consists of primary forest composed of shrubs, thick tree canopy and bamboos, having an elevation of 557 with a surrounding ambience of recorded 20 degrees centigrade and 76% relative humidity. An Ichthyophis multicolor, a limbless amphibian, Sicilian. So we have collected altogether around 7 samples, and out of which 3 were live samples and 4 were roadkill. As it can be seen in the slide, these are the rod connecting the border, the lower side with the lesser canopy are the buffer, and the upper side with the thick shrubs, with the thick trees are the core area. And here we have the plantations of olive, then below here a rubber. So it's found in the mostly in the buffer area. Then it has got a snout vent length measured between 305 to 443 millimeter. We encountered the species of this eye multicolor along the road, dividing core and buffer in a vicinity of a village, a fringe village called Lallen in Puldungsei range. And it was mostly observed in the evening between 6 to 9 o'clock. It was very recently collected on 30th of October to 2nd November. And the surrounding habitat where it was collected was mostly of mixed secondary forest, having teak, then betel nut, then rubber and olive plantations, tall grasses, shrubs and bamboos, and it has got an elevation between 659 to 797, a little bit higher, twice higher than where we collected all the frogs. Now, Buphoides megalayanus, this was for the first time described by Yazdani and Chanda, 1971. Since then, this has been vaguely reported from Meghalaya in Cherrapunji. Now, this is the proof of where we have collected from Dampa Tiger Reserve. And now we have all the molecular data with an accession number. So this would be the confirmed report from Dampa Tiger Reserve as well from Mizoram, not only in Meghalaya. So there were 2 specimens of Buphoides megalayanus encountered in Dampa Tiger Reserve. It was way back in 2001 reported as undescribed by Pawar and Biran, but now this has been confirmed through taxonomic and genetic characterizations. Now, due to in Meghalaya, this particular species, Buphoides megalayanus, due to population and the habitat under continuing decline because of the rock blasting quarry, and hence this species is listed critically endangered. And the species was, the individual was collected on the rock fissure at Teire Forest, having an elevation of 296, much lower than the previous samples collected. And the area is covered with primary forest composed of woody lianas, shrubs and bamboos. So one common characters about all these amphibians are they were collected near mostly there where there is a lot of woods, trees, shrubs are available, not in an open area. Now moving on to Buphoides kempi. It is diagnosed by small to moderate body size with a minimum of 17 to a maximum of 29 millimeter. And it can be easily identified by the presence of dorsum. When observed closely, there is a granular with small conical warts scattered throughout, which gets more intense towards lateral area than the dorsal region. In a study, the only male individual measured 26 millimeter. And this species is categorized as data deficient. So far, there is least known description besides the Garo Hills of the Northeast. In our study, the specimen was spotted. The individual was spotted on the rock crevices. And one thing which is very surprising is that not only this particular thought was found, but along with one scorpions, Euscorpion species, which is not that far away from that Kotlabung Fall, a waterfall, a high waterfall, which is about 150 feet high. So that was in Puldungsei area, having an elevation of 769 meter. And the habitat is moist forest composed of shrubs and bamboos. So we have the live as well as the preserved samples. And upper provides the area where they were collected. Now coming to the main point, threats and the conservation challenges. Extensive practices of shifting cultivation. Right now the season, November has started now. By as early as beginning of January next year to March, April, it will continue for 3-4 months of extensive deforestation in the buffer areas, and then they'll start burning it for shifting cultivation or jump cultivation, which is quite commonly practiced. And not only that, collection of fuel wood, indirect drivers such as excessive exploitation for NTFPs, non-timber forest products, monoculture plantation, road kills, use of pesticides, forest fires, consumption and sell at local level, lack of knowledge and awareness were creating impending crisis for the Dampa Tiger Reserve amphibian diversity. And some of the field observed are listed under. The most common one is the deforestation, logging, then forest fires, which is quite common in the dry season from January, as early as to January to April. Dependency on forest resources such as uprooting bamboo shoots, which is quite common during rainy season, June, July, August. Then over harvesting of all these animals for food, for selling at the local market. Then road kills, which is quite easily observed here in the slides. Then lack of awareness. Although this year we are involved in a project, we have conducted 3 times awareness program, which we think would be helpful for the local, and they have also shared their interest and their curiosity with us, but more have to be done on this context. Now, moving on, discussion and conclusion. The largest protected areas altogether, Mizoram state has got 10 protected areas, of which Dampa Tiger Reserve is the largest. Now it harbors, now it roughly gives us the idea of how much amphibians have been directly infected. This is based on the context of the Dampa Tiger Reserve. Now it harbors diverse groups of herpetofauna. Roughly we can come to know about it. Now among those, the recorded 35 amphibians. Earlier, Pawar and Biran, it was 18. Then 2021, it was increased to 28. Now 7 more, 35. So total we have around 35 amphibian species recorded so far from Mizoram. Now among those, 1 species is categorized endangered, 8 species as data deficient, 2 vulnerable, 14 least concerned, and 10 species not assessed as per the IUCN 2020. Now all these, not only the amphibians, they need a protection by means of effective education and control of pesticides. As we have seen in the previous slides, there is a lot of slash, burning and uprooting, exploitation. Now during those processes, in order to produce or get a good harvest, there is a common practice of using a lot of weedicides, pesticides, and all those offshoots would go to the nearby streams, so which would further on pollute. And then all those running streams, waters now would be made use for a domestic purpose, so which in turn affects the health in the local area. Management of waste disposal is a much solid, as well as waste management is a need of the hour. Now, significant proportions of amphibian diversity of Southeast Asia, including Dampa Tiger Reserve, is likely to be hidden. So it's a research hub area. Now, people have started getting aware of the location of this Dampa Tiger Reserve, where especially in terms of research, this is a research hub area, because it's as we have seen, morphologically cryptic and misidentification between closely resembling species such as Rhocestes manipurensis and Rhocestes rejakhani. Just at the outer appearance, morphologically, it looks similar, but however, when observed closely based on the molecular data, it's completely a difference. Similarly, with Hoplobatrachus tigrinus and Hoplobatrachus littoralis, Microhyla muclesuri, and Miclohara mimigensis, Fajarvaria multistriata, and Minervaria asmati, which are quite common in the region. Hence, the importance of inventory studies and role of systematics, molecular taxonomy, and further exploration and reporting new species is the best option. Now, anthropogenic sites hassle with discovery of some of the most resilient amphibian species. Under presentation in amphibians, especially these range restricted species for conservation decisions involving protected areas is crucial that needs wider approaches from stakeholders and policymakers. Then amphibian shows high degree of sensitivity towards alteration of habitats. Such field-based surveys are important to provide the insight about community organization, persistence across different sections of forest recovery. Not only will it provide fundamental baseline for conservation management, but moreover research in batra co-diversity as well. So these are 7 different published papers based on Dampa tiger. So the first one was in 2000, the twin spotted tree frog, which was not confirmed but reported based on the secondary information by Pawar and Biran. We have confirmed it. Then the next, the reason why we have selected this topic was based on the second point, an annotated checklist of amphibians in and around Dampa Tiger Reserve. Now, these are the further published papers on different journals. Now, an acknowledgement from the Chief Wildlife Warden for providing us a permit, then the funding agencies, the Forest Department of Mizoram, then NMHS, Department of Biotechnology. So, thank you. Any question, I would be happy to receive it.
Thank you, sir, for your extensive presentation. Over to Ganesh, sir.
1 second. Mr. Simpson, can you hear me?
Yes, sir.
Right. Despite all the threats and challenges that you had listed, do you think your updated checklist of amphibians in Dampa Tiger Reserve is on the encouraging or disturbing trend in terms of species diversity and species abundance?
Sir, this would be a cross-check as well as an updated of the amphibians.
Okay. You said 35 species have been checklisted.
Yes, sir.
What about any previous records? Was there any more amphibians or lesser than these?
Prior to Pawar and Biran 2001, there was no such documentation or any evidence of published publications. So
Okay.
after his publication report of Dampa tiger, not only Dampa tiger, he did an extensive and very good amphibian studies in the whole Mizoram. But so far we have selected this Dampa tiger because it harbors the diverse herpetofauna among the 10 protected areas. So considering that point, and as well as this is the second from the Tiger Reserve.
So you say that the checklist is an encouraging trend.
Yes, sir. That would be the appropriate one, sir.
Okay. Thank you very much.
Thank you.
Thank
That's
you,
all.
sir.
Thank you.
Sorry to interrupt. I want to take the permission of the chairpersons, Dr. Ganesh and Professor Pratibha. Just a request. We have
Yes.
3 and some more getting added oral presentations.
Okay, sir.
We had to start a bit late, but I thought we will go ahead with the keynote speaker's address. Dr. Pratyush has joined us. So even if the oral presenters, with your permission again, we will have the keynote address of Dr. Pratyush and then continue with the oral presentations. Will that be okay, Ganesh sir and Pratibha ma'am?
Sure,
Yes,
sir.
sir. Yes.
That's
Go ahead.
starting with Dr. Pratyush. The oral presenters, sorry for this. We will give you ample time. We are not restricting you to 5+2. We are allowing you more than that to encourage your presentation. So we will now hear it from Dr. Pratyush. We have the next session on that. I now, before that, I would call upon these 3 people. This is Vanlal, Gospel, Yagnapati. 3 of them are pending presentation. Anybody else has a presentation pending? I see Mr. Narendra Sahu, who could not present yesterday. Sir, will you be doing it today? Okay, no response. So there are 3 pending. We will start with the next session. I now call upon Dr. John Paul, Assistant Professor from the Department of Zoology, St. Joseph's College, to introduce our keynote speaker for today. Over to Dr. John Paul.
Good evening, everyone. Welcome to the second day of the Herpetology webinar. I'm here to introduce the keynote speaker of the day, Dr. Pratyush Mohapatra, Scientist D, Zoological Survey of India, Central Zone Regional Center, Madhya Pradesh. We heartily welcome you, sir, to the SJC family. And to introduce about Dr. Pratyush, he has earned his PhD from the North Orissa University on the topic of systematics and biogeography of snakes of Eastern Ghat ranges of Orissa. His fields of specialization includes herpetology, animal taxonomy, multi-taxa biodiversity survey, wildlife biology, community conservation, human wildlife conflict mitigation, wildlife management in protected areas, and he is honored with several awards, including those of Snake Conservation Award by the Orissa Environmental Society, the Charles Stevens Grant in Aid for Herpetological Research from the California Academy of Sciences, and many others to be pointed out. And Opilian Indiacaris Pratyushi is named after him. We congratulate you for that, sir, for your wonderful contribution to the herpetological work. And he is also involved in describing several new reptilian, amphibian, as well as cicada species. He was the lead author for the guidelines on mitigating the human-snake conflict, as well as he was a key member on the project of human-wildlife conflict mitigation of India, and the project undertaken by the Ministry of Environment and Forest and Climate Change, Government of India. He is currently working on several government funded projects as principal investigator and co-principal investigator. He is also the author of several articles, chapters, and books of various topics pertaining especially to the herpetology. He is a member of several committees, including that of the IUCN Red List for reptiles of South Asia, Amphibian Arc Conversation, needs assessments for the amphibians of India, the IUCN Viper Specialist Group to I mention a few. We are overwhelmed with joy in welcoming you, Dr. Pratyush. Over to you now for your session.
Thank you, Dr. John. Hi, am I audible?
Yes, sir. Yes, sir. Yes, sir. We can hear you.
Okay. So I'm actually in a low network area, so I'll switch off my video while presenting. And thank you for warm welcome and respected dignitaries here, and Jai especially, and my colleagues. I can see Dinesh over there and many other friends, and my dear researcher friends. So today I will be talking about reptiles, and I'll try to motivate you to take reptiles as a study subject. And like this is just like I'll try to motivate you to take up reptile as your professional career. So before starting that, I would like to say that I'm interested in reptiles when I was a child, like when I was a kid. So since then, I was fascinated with snakes, especially, and like many other friends I have. So many of them are become professional herpetologists in latter part of their career. But like to stick to your hobbies and to make it a career is really challenging, and you have to do hard work for this, and definitely you have to keep up your passion, and then you have to study in right direction so that you achieve your goal. So thankful to God because I'm one of them, like who carried out my hobbies as my profession. So now I'm working as a scientist in Zoological Survey of India, as already mentioned by Dr. John, and I am pursuing on reptile taxonomy, conservation, and all these things. Apart from that, I also study on scorpions, and apart from that, like most importantly, being in government, like we take part in several like policy level decisions, and based on our experiences in this field, so we try to give some reasonable thoughts about all these things to include like especially reptile conservation into policies and all. So what basically I want from the students, like nowadays students are very smart and they are pursuing like from different fields of science or art subject. They're also interested to pursue herpetology, and we have seen many very good taxonomists which are not even from zoology. So they are doing excellent taxonomic work, and they are also like now people from the field of zoology, those who have studied animals, and they are also doing good work. So herpetology has no barrier. If you want to be a herpetologist, like generally, uh, apart from zoology, other fields, people they also can become herpetologists, but it is little tough. Like if you want to be settled and if you want to be like to pursue your career in herpetology and academics or in research, so it becomes little difficult. So I'll try to uh, make you a herpetology, a professional herpetology by yeah. So I think in this forum, like whoever is there, so every one of you is a budding herpetologist or you are already interested in herpetology. So we need also lots of students to join this field because we know very little about different groups of animals. Just any example you can say, like for example, a common species of snake, maybe we know very little about them. And when it comes to conserve them, or like we have seen in IUCN assessments, like what on many species of like reptiles, especially many common ones, we have very little knowledge about their ecology, about the biology. So all those studies, earlier, like before, maybe before, like in 70s, 80s, people were writing lots of things about naturalist information, whatever they were observing, they're putting them into pen and paper. But nowadays, the trend is little different. Now we are much into other aspects, like the true naturalist approach. It's somehow missing. I don't know why, but still, like we have many things to report because at the end of the like day, when we are assessing a species or we want to conserve the species, we have very little knowledge. So every knowledge on reptiles or amphibians or, for instance, any other animal is really valuable when we try to conserve them. In this presentation, I'll be talking about the historical perspectives, like people who worked on different, they showed us different, like in the taxonomist, basically in pre-independence era, and after that, like what trends of like taxonomists and recent prospects in taxonomic studies. then all these taxonomic works, how we do taxonomy, then conservation and ecological works, and there's so many fields. So in herpetology, if somebody wants to like enter into this field, so the scope is very vast. Like you can choose any subject, like you can be a taxonomist, you can be a conservationist, you can do lots of ecological works, which will be very important in future times, because when we are talking about climate change and all, we need ecological data for this climate models and all, like to save species. And now venomics, like in India also we have an established lab in ISC who is exclusively doing work on venomics in a much advanced level. So we have lots of things coming up in terms of venomic studies and developmental biology. Even in India, we have a few universities who are working on developmental biology in reptiles, especially regeneration studies, stem cell studies, teratogenesis, all these things. In few universities, they are working. So as I am a taxonomist, I'll be trying to motivate you to become a taxonomist, because we know like we are in search of people or good taxonomists who can be useful in studying animals. Till now, we don't know how many species we have. Like the number is ever increasing, and also we have seen there are like lots of species which are getting new names. So all these things, like we need to work seriously. We need some serious taxonomists to do justice to taxonomy, especially. So I'll talk about what is taxonomy. So apart from that, taxonomy is such a field in which it involves several things. Like if you love to go out, so taxonomy is a field where you can do field surveys. You go to the forest, you find species, like you find different animals, so they're cryptic animals, so to find them. Then if you love sitting in museums and study animals, so this is also a field where you have to compare different reptiles which are housed in different museums. So you study the museum specimens, and then of course you
Sir, Dr.
yes.
Sir,
Pratyush, sorry
excuse,
to
yeah,
Sorry.
interrupt. So the
Yeah.
slides are
yes,
not being
slide, yeah,
shared.
Okay.
yeah.
Is it
We're just
moving?
continuously talking, couldn't disturb you. And the slide
Oh,
is not
oh,
being shared, sir. Please just check it out.
not being shared. Okay, sorry.
Yeah, it was there, sir, beginning. Now only,
Okay,
in between only it got cut off. Just check out.
okay. I'm sorry.
Yes, sir, yes, sir, yes. No problem.
Now it is visible.
Yes, sir, yes, sir. Now it's okay. Yeah, now it's visible.
Okay, okay. Sorry. So I'm trying to say that doing taxonomy is all fun, because you will be going to field to find animals, like you'll be studying museum specimens, and it is just like a detective science. Like once you get an animal, like get a reptile, so you have to match it with the previously known species, and then if it gets identified, like based on the characters, if you find it, okay, this is species A, so then your job is done. Otherwise, like in taxonomy also, it answer important biological questions, like how this species has evolved, and what are the special requirement of this species, and so many other biological questions, especially evolutionary aspect you can also study, and so many things. And it is a perfect balance between field and lab. And taxonomy never gets old, because every time you have something new which is coming up, so it is not never gets old or never gets saturated. And doing taxonomy means you have to refer to the like Bible of taxonomy, which is the International Code of Zoological Nomenclature. So which it has several sections, and you have to follow the sections when you are naming a species. Like I'll be talking a little about this ICZN and different like codes of it. So because if you want to be a taxonomist, so sometimes you have to follow the code. Like sometimes you get stuck in some like critical questions in which you have to follow several codes, and you have some articles, and then you can like name the species. So systematics is a broader field which is study of biological diversity and evolutionary relationship among the organisms, and taxonomy is a branch of systematics, so in which you identify a specimen, you classify it and you fit it into different hierarchical classification hierarchy and the nomenclature or the name part of it. So when we get a specimen, like to identify it, sometimes like we get excited, like we got a new species or something like that. But sometimes we do like morphological study, we do genetics of it, and we are convinced that this is a new species maybe. So, but in most cases, what we generally lack, like what we see in recent trends, so people often neglect the nomenclature part of it. So the nomenclature means being a taxonomist, the name is our like priority. So we need to give justice to a name. So in some species, we often ignore the earlier available names. So that is a problem. So there are so many critical issues coming up in modern taxonomy. Like in recent years, we have seen in many cases, researchers often ignore the nomenclature part of the species. So in that way, like it is very difficult to again reassess the species name. So that is what I'll be giving you some examples, and it has happened in past. It is also happening right now. So before going into taxonomy, so we all know like species concept and all. I'm not getting deep into it. So like what is a species and all these things. And like there are several species concept also. Like mostly we follow the biological species concept and the like phylogenetic species concept. So biological species concept has certain limitations, but recently like we all follow the phylogenetic or evolutionary species concept. So and there are different kinds of species or terms related to species. For example, people are talking about cryptic species nowadays too much. So it is which are morphologically similar but genetically they are distinct species. So in that way, like a taxonomist has to like take different different parameters into consideration. For example, how they are like biogeographically they are disjunct, or so many things like we include to define a cryptic species. So they actually morphologically they look very similar. So based on morphological characters, you cannot identify them. But if we cannot identify them morphologically, then what is the necessity of describing it as a species? So in that way, like I'll give you some examples. There are several interesting case studies nowadays also, like why we should consider it as cryptic species. So in herpetological studies, so like as I said, like if a species is geographically disjunct, so in that way they cannot mate with each other. They are already they are separated million years ago, and they have developed certain kind of barriers like that. That's why they cannot mate with each other. They cannot like produce, like then the biological species concept comes into existence. Allopatry, like all these things will come into existence. Then comes sympatric species. So many times, like two species, they're sharing the same habitat or same range. Then allopatric species, like inhabiting completely different geographical area, and there are many chances that in future there will be coming up as different two distinct species. So like that, there are several species related terms. Another one is subspecies, so which was used earlier, like 20, 30 years back, like people were sticking to subspecies, but these are the lowest taxonomically nameable category. So nowadays people prefer to stick up to species level. And in that way, like many of the subspecies are being resurrected as distinct species. And before going into the taxonomy, like you need to know about holotype, paratypes, syntypes, all these type related names. So if you have questions, and we can discuss about it. So basically we have like holotype is a single specimen which is used by the author and designated as a type. So nowadays, without holotype, you cannot describe a species. So recently, we had a problem like in from there was a new species of crate described recently last year, this year maybe. So which doesn't have a holotype. So the name didn't become valid. So that was Bungarus Whitakeri or something Romulus. It was named after Romulus Whitaker. So anyway, so in that case, like there's some problem if you nowadays if you don't have a holotype, you cannot designate a species. But earlier, like before 1961 up to or before that even, like there were species which were only based on figures or drawings, like those are also considered as holotype, or those are typically called as iconotype. So based on a drawing, a species has been described. Apart from that, holotype, paratype is there, the specimen cited in the original description other than the holotype. Similarly, syntypes and lectotypes. The neotype, if the holotype is lost, then it has been replaced by another type specimens, which is called as neotype. Then topotypes, it is also very important term, because many a times if there is no holotype, and it has been described based on iconotypes. So in that case, like topotypic animals are of very important, because for genetic studies and all, you have to collect the topotype animal and then you can do the genetic study. So we all know about Linnaeus, who is the father of taxonomy, and some of the species described by him, which are distributed in India, or some species are given. Then Legends of Indian Herpetology. So these are people who contributed maximum during pre-independence, like in late 19th century till early 20th century. So of which Russell is very like, I like him because he is the father of Indian ophiology, and he has not described, but he has written 2 volumes of book on Indian snakes. So he was a surgeon basically. So he has done lots of experiment on snake bite and all. He was posted in Visakhapatnam, but he had several collections from different parts of India and Southeast Asia. So in his book on snakes, on collection of snakes from Coromandel, so he has mentioned about several specimens. He himself described only one species, that is a valid, like that is red sand boa or Eryx jonai. But based on his painting, many snakes have been described. So apart from him, Gunther, he has a great contribution in herpetology. Then Bollinger, then Frank Wall, who was he had written several stories about snakes, lots of naturalist information. He has also described several species of snakes. Then Jedon, Bedom, so all these people, and finally Malcolm Smith, so who has written the reptiles of fauna of India. And these are all people who has contributed tremendously in herpetology, in reptile taxonomy, basically. So here I would like to point out one interesting taxonomic case study. So in which this year we published one article. It was about some more than 200 years, like one species of snake has been named in a different, like it was means the type specimen of the snake or the iconotype was actually belonging to another species. So it was a very complex taxonomic story. So this year we just tried to clarify after like 5-6 years of digging literature and all these. So this is about Lycodon, one species of wolf snake. So actually the type specimen belonged to like this banded racer. So Argyrogyna fasciolata is the name of the banded racer. So this name fasciolatus has been shifted to Lycodon. So these two snakes, though like in live condition you find no differences, but in the picture, in the iconotype, it was a problem, like it was named differently. The name was with another species. Anyway, so this is how, like, lots sometimes like you get into taxonomic problems, this problematic situations in which names have been exchanged or people have described it in another name, and then you have to search for a suitable name for the species, and then like you have to name it. So well, so this is how we sometimes the complex taxonomic questions arises and we have to solve it. So before that, like collection of reptiles, how we do, how to find them. So all these things, you if you like are associated with some herpetologists, you might be knowing. But even field work is a very tricky business, and it needs like lots of energy and passion, and like your enthusiasm should be like it should be a kind of like a passionate thing. So you have to be prepared for field surveys, and nowadays permits, like getting wildlife like permission to collect specimens or collect like different scheduled species is also a bit difficult. And then you have to have collection permit if you are dealing with scheduled animal. Then how to find specimens, so it needs lots of skills, and you have to develop a search image. It also develops along with your experience in the field. Then catching or collection, sometimes you can find a species, and it becomes difficult to collect it because it will run away. uh, Reptiles are, anyway, they are cryptic, so you find them very rarely in some specimens, and after you see them also it becomes difficult. So in that way you have to be sportive, like you have to jump, you have to dive, sometimes you have to like climb trees, jump from trees, so many things, crazy things you have to do while like catching specimens. And after collecting the specimen, if you are studying it for taxonomic study, sometimes you have to preserve the specimen, you have to sacrifice the specimen. So preservation is also very, very important, and it takes lots of time to preserve a specimen properly, so that like all the taxonomic characters are intact and you get to collect data from it. Then you have to catalog the specimen, like with all the metadata. And like to find reptiles, like there are several methods, like you can actively search for the animals, like you have to go to the microhabitat, you have to find them, and then hand pick it. Otherwise, like you have to sometimes like use different tools to trap them. A pitfall trap is also another interesting, like important thing, like you can do in field. You can try for skinks and other animals which are difficult to catch sometimes. Then use of boards for arboreal species. So there are different, these are also useful for ecological studies and reptiles. Catapult, I have used it for like arboreal species, so it also works well. Netting, glue traps, all these are different methods to catch reptiles depending on which group of reptile you are trying to catch. And snakes, especially, you have to be very, very careful while handling snakes, and especially when you are in like dense forest or unapproachable areas, so in which like the chances of getting hospital is low. So in that case, you have to be very, very careful. You have to follow all the safety protocol before like catching or handling any dangerous reptiles, especially the snakes. So you have to be like equipped with like these tools like snake hook, tongs, and other things. Like if you are going to catch snakes, and there are several like hook bagging methods. So, and for this you have to be careful. So then in reptiles, like while studying them, after you fix the specimens, then you need to study them. You have to find out characters, how you're going to like fix it into a this particular species, or like match it with different closely related species. So you have to take into consider several macroscopic, microscopic, or molecular tools. So like macroscopic characters which you can see like with your naked eyes. Microscopic, sometimes like you have to find like some micro ornamentation on scales, or sometimes like some internal structures you want to see, all these things, so which provide the variation in which potentially provide phylogenetic information. So this is as per definition. So any character which has certain importance in taxonomy, so those can be called as taxonomic characters. Sometimes in some species, like adults, they look similar, but in juvenile stage, so they're contrastingly different. So in this case, there are two species. Like the top one is the Hemiphilodactylus aurantiacus, and below is the Hemiphilodactylus minimus. So when they are adult, because they changed color, so they look almost similar. But in juvenile, so you can see these patterns, they are almost constant in juvenile, like till they become attain adulthood. Then methodology for studying specimens. So preserving specimens means fixation. Generally, animals are fixed in formaldehyde solution, 4 to 5% formaldehyde solution. And before that, you of course need to take tissue out of it, and tagging, storage, photography, all these things are done. And so nowadays the craze for new species is too high. So people often like they maintain secrecy while collecting specimens, and they there are like lots of competition in describing new species and all. So that's going on. So these are some of the morphometric meristic characters I was talking about, like all these standard characters you can take, and this is not possible like in this forum to deal individually with each characters, but there are some like common terms you can easily correlate with the like snake body kind of thing, like head length, head width, frontal length. Like these are different scales named after their position: frontal scale, prefrontal, supraocular, parietal, and all these head scalation, their measurements are very important in identifying a species. So, and these are different names. And to count the ventral scales in snakes, we have also standard protocol as per Dowling 1951. We follow this invariably to count the ventrals, and in that way, like we have like preventrals and ventrals. All these characters are taken into consideration. Similarly, in geckos, like we have several taxonomic characters, taxonomically important characters, like lamellae count, and then all these measurements, then pre-cloacal femoral pores. So all these characters are taken into consideration. All these morphological characters are important in separating two different species. Similarly, in turtles, there are different morphological, morphometric character. Apart from coloration and other things are also there, so which are taken into consideration while describing species. In skinks, similarly, different characters are taken into consideration. So apart from all these, like all these morphological characters, now people are also following different anatomical characters. For example, hemipenial morphology and reptiles and snakes. So these are like forgotten field, like forgotten characters in taxonomy. I would rather I'll be saying, because earlier these were used as tools. Like some people, there, some researchers or scientists, they have tried to uh, uh, provide information on these anatomical characters, but now maybe we don't have much time to deal with all these things because of too high competition in this field. But you can slowly study them. These are very interesting characters, anatomical characters especially, which gives you idea about the evolution of the species. So, and I have here shown like the proper way to preserve a specimen, like if it is a male, so you can avert the hemi penis, which can be studied later. And these are information on the species, from where it was collected, and geo coordinates and all. And here in this picture, I have shown you a hemi penis of Rhapa guentheri. This is a skink, and in which like I got fascinated to study it because I read one paper in which like I basically study the hemipenis of snakes in detail. So in this, there was a paper on limblessness associated with hemipenis evolution kind of thing. So I also found this Rhapa guentheri is one of the limbless, like near limbless, like it has very short limbs and it has very long body, and how these Rhapa skinks, how they manage to mate with each other. So there are certain kind of, like you can see in this, mushroom shaped hemipenis, so which helps in supporting like in sperm transfer. Like these are special modifications in skink. I didn't find in other skinks especially. Sorry. So and yeah, so these are some of the preparations. So apart from this, like in scale reduction, like in snakes especially, so when we are talking about scale round the body, we generally follow three distinct regions, like one head length behind the head at mid body and one head length before the vent. So at these three positions, generally we take the scale around the body counts. But apart from that, like there are certain taxonomists like Clark and Inger, they proposed the scale reduction, because snake has both side tapering body, so in which like the scale around the body varies at different positions, and these in some species are also key characters to distinguish them from another species. So it is a very tedious process though, so which also gives a pattern, like in males and females it may be different, but it can be a species specific character in some species. So these are very tedious way, like when you have lots of specimens. So this is a way how to give scale reduction. It has a formula. So left side and right side of it, how to write. So these are practically when somebody wants to go deep into it, so you can study it, and there are standard methods to write the formula. And if you have large sample size, so you can also concise it and write it in this way. You can follow some papers, it has been given. So then nowadays, like we follow the integrative taxonomy, like you can see some classic works in integrative taxonomy in reptiles is basically like just to cite few, like Deepak et al. In 2016, like from earlier we had one Citana ponticeriana fan throated lizard, and how diversified, like how diversified this species was. So he basically, the group told us in that by integrative taxonomy, by doing proper integrative taxonomy, like wide sampling through almost throughout the India, and different characters, morphological as well as molecular character, to describe species. So many a times also the taxonomic validity of species, like which is done like in some groups, and mostly in the agamid lizards, and in Cytopodian, Ishan Agarwal and team, like they have also provided lots of interesting findings in the Cytopodian group, even Hemiphilodactylus and so many other groups of geckos dealt by Ishan Agarwal, so and Bharat Giri and so many other friends. So those are very important insight into the taxonomy of the species. Then I was talking about like cryptic species, so how morphologically, although they look similar, or sometimes genetically, these species, like in Lycodon, somebody has to work on the Lycodon, because Lycodon olicus, this is one of the common species found like throughout India and adjacent, like in Southeast Asia and some parts. So this species is so widely distributed, but it has several morphs. But people are yet like trying to study on the cryptic diversity in this one species. So it again it be coming up and people are trying to segregate this Lycodon olicus into different species. And recently, for example, king cobra study by Gauri Sankar, so the taxonomy of king cobra, so in which, like earlier it was monotypic and one species distributed in India and Southeast Asia, many countries of Southeast Asia. So now how different, uh, different it has, like Western Ghat has, we have one species, and Eastern Ghat and Indo-Chinese, and different clades of king cobra has come into existence. And so these are all like dealing with cryptic species. You have to deal with the taxonomy, and you have to deal with the phylogeny of species, and then you have to delimit species based on all these things. So integrated taxonomy, basically it is a science that aims to delimit the units of life diversity from multiple and complementary perspectives. So it takes into consideration multiple lines of evidence to delimit a species, to delineate a species. And you can put anything, any data available on species. For example, geography, as I was talking about, the barriers, different barriers, and ecology, behavior, morphology, genetics, so all mixed together when you are describing a species. So it going to be a like true justification for that species. Otherwise, like many a times, shallow description, or so all these leads to synonymizing the species in future. But above all, again, taxonomy needs to be considered, because if you do not consider taxonomy or earlier available names, so then also describing new species doesn't it may attract like synonymization in future. So I'll just talk about Hemi Penis. I work on this especially like to know about different aspects of the evolution. So in snakes, it is a very elaborative structure, as because snakes do not have limbs. So with the Hemi Penis helps them in like anchoring into the female genitalia and for secure sperm transfer. So in which like many snakes they have developed spines and they have different kinds of micro ornamentation, so which helps them to anchor into the female genitalia. And many a times, like we often, while pressing the tail base, we see the hemipenis getting popped up, and but that is not the full structure. So when the snake is mating, so it averts the hemipenis fully, and so all the many characters are there towards the distal portion of the hemipenis, like away from the tail base. So like this, like when you study the hemipenis of a snake, so it gives you lots of character. So which may be species specific in some cases, and also these are evolutionary very important characters, because when we talk about biological species concept, so this is one kind of like pre-zygotic barrier. And the structures are variable in different species. For example, in Dendrolaphis, if you see, so in one genus, this much of diversity. So this is Dendrolaphis humayuni hemipenis, and this is Dendrolaphis andamanensis hemipenis. So see how different they are. So it shows that these two groups might be of two different clade, which has been like getting support from the genetic data as well. So like this, so many things you get insight from these anatomical structures, and you can see the diversity of hemipenis in different groups. And each family is like has some typical characters. For example, colubrids generally have a single hemipenis. It is not divided or bifurcated. And all these elapids, they have bifurcated hemipenis. So here you can see Naja Naja. Bungarus fasciatus, it has also like the Sulcus formaticus is divided like that. So some typical characters are there for different families. These are like Homalopsid, Cerebrus, Rinchops, and like that. So in many different families, they have uh, some typical characters in the gross hemipenial morphology. So then the teeth structure. So that is also very important in some species. The number and shape of teeth, especially the maxillary teeth, are of importance. Mandibular teeth as well. So the internal structures are also important in terms of taxonomy while we describe. So and giving like these skeletal characters is actually uh, uh, we should actually give all these skeletal characters, because it also helps when we are like um, for these paleontological studies, especially when people are getting any kind of fossil or something. So actually being like vertebrate taxonomist, we should also provide information on these extant species, so that like we can means corroborate with the fossil data, like to get much deeper insight into the evolution of species. So yeah, so these are the things like people can do. People can take up all these studies. Students can show interest if they show interest in these groups. So these are classical taxonomy. Actually, these are not new fields, but because of difficulties in studying them and the tedious work it involves, so people often ignore these fields. So nowadays, these micro CT scans also proved highly beneficial, and without like tampering the specimen, you can also study them. But these are little expensive, but otherwise, like these also provide lots of insight into the basic like the structure, the skeletal structure of different animals. So in people are nowadays people are providing lots of information about by doing micro CT scans, and you can and it has also several characters which can be applied to taxonomic studies. And then comes photography, so which is also very important. Like people often like uh, taking good photography is again an art, and like you have to learn photography, basic photography, so that while you are presenting or when you are like writing papers, so you need good photographs to be presented to, which shows different diagnostic characters of a species. So, and this is a better way of presentation. And for specimens also, you have to take pictures like which shows key characters, like whatever characters you are putting in diagnosis, all those characters should be visible when you are presenting your specimen information. So it should be of professional quality, and so that like it'll be easily understandable. Then tissue samples for genetic studies, like like we all know, like from blood, muscle tissue, liver tissue, we can take which are rich source of DNA, and you have to put them in vial in absolute alcohol and store them, and then you can go for DNA extraction, amplification, and all these things, and genetic studies. And many a times in taxonomy, people do mistakes without like they ignore sometimes the available names, or they like they become overconfident while the, they get over enthusiastic while describing species without going into the history of a species. So often these fall into like synonymization category, so like that. Then when you get specimens, so you need scientific institutions where you can deposit your specimen. So then Zoological Survey of India, Bombay Naturalistic Society, NCBS, and many museums are also there where you can deposit your specimens. But it is always advisable to store your specimens or deposit your specimens in some national repositories, which which will be taking care of your specimens, and they are doing that. That is the job. So like JSI and all its branches, all its regional centers, they are also national repository. So as Bombay Naturalistic Society, even WI, NCBS, so they have all storage facilities where your specimens will be securely stored. Then yeah, so these are all about taxonomy, and in ecology also we have very little work on reptile ecology actually. So Dr. Karthikeyan Vasudevan, Hari Shankar, and Hari Krishnan, sorry, and they have also worked on the ecology, like some like in Andaman Nicobars, and Dr. Karthikeyan Vasudevan worked in Western Ghats. Some ecological studies we have in India, but we need lot many works on ecology, especially many common species. We do not have any kind of ecological data. Even in Agumbe in Forest Research Center, they have also tried doing some ecological work on king cobra, and these are all necessary in future times, because when we think about conservation of a species, we need to have some ecological information on these species. And apart from that, like some behavioral studies, very nice works has been done by IISc. So those are also very important in terms of knowing species in much detail. And then the conservation of reptiles, we have like in our country, we have Wildlife Protection Act, in which like there are 18 species of reptiles in Schedule 1, Part 2, and 7 species in Schedule 2, Part 2. So these are the species which are like given higher priority in terms of like legal protection. And Now like in near future also there is a amendment coming up, in which like many species are included into the list, and some of the common species are removed. So in that way, like Wildlife Protection Act is getting amended. And there is also one IUCN. Like IUCN's main job is to like do global assessment for species, in which like they have all these 7 categories, like critically endangered, vulnerable, data deficient, and like these are threatened category up to vulnerable. So like that, like they put species into different categories based on the present like the available literature. And that is also like this IUCN exercise is also helpful in getting funding, because many funding agencies, they provide funds for the threatened category of species. So in that way, like if a species is in critically endangered or endangered category, in those species you will get good amount of grants to study. So for researchers also, it is very important, like while choosing, like you can prioritize these species to take up ecological or conservation related works. Then uh, although in terms of opportunities, herpetologists are like much like they are way back in terms of like people working on tigers and elephants and big animals, but still like there are lots of scopes, and nowadays funding agencies are also understanding the importance of these little charismatic species like snakes or like crocodiles, turtles, so like that. And in India, for example, like turtles, sea turtles especially, so on which many works have been done. So those are charismatic species in the reptile groups, and they get lots of funding to do research on sea turtles. But on the other hand, like getting fund for freshwater turtles is little difficult. So in that way, like you can avail funds from local forest department, even biodiversity boards, or like some other funding agencies if you want to work on conservation of these reptiles, which are not so charismatic than tigers, elephants, etc. And then what we basically do, like we try to translate our taxonomic knowledge into species conservation through various means. And so this also helps like by through doing status surveys for species, then in some species recovery programs in situ or ex situ in reptiles, and all like and species management, preparedness and management of exotic reptiles. So nowadays it will be a future topic, like when already India has lots of exotic reptiles, and once they are like, we have also reports that already there's many species are already like released into wild, and they are slowly colonizing. So these are all big tasks for you all, like to tackle this problem in future. So we have to be very careful, and maybe in near future strong laws will come to combat these kind of situations. For example, if any snake is released into a like exotic species of snake is entering into a natural habitat, and they start breeding and they start like the population increases, so it may be a big problem for the native species of birds or other reptiles. So we have lots of examples of these invasive species in Australia, for example, Boiga irregularis, then python bivitters in the US and all. So people are like, government is spending lots of money controlling their population. So we don't want such kind of situation in India, but let's see like how government is taking up initiative and combating this exotic reptile trade. Then in taxonomy research also very less funding opportunities, and now sometimes it takes long time to understand the taxonomic problem. There is competition to publish first, and there are lots of groupism. So you should not fall into all these things. But if you do good work and you collaborate with good people, so of course there are like lots of good things for you. And there is vast scope to work on taxonomy, because we have still lots of new species are coming up, and like in some groups, like these are almost untouched. If you start working on them, they are widely distributed species, but if you start working on them, start collaborate with people from different areas, and good people, you can have collaboration, so who have access to museums. And so in that way, like a team work, you can also do lots of wonderful contribution to this taxonomy or to the science. And with increasing number of snake lovers and rescuers, so it is again a good thing. On the other hand, it is also bad thing, because lots of not so good people, they are into the field, and rather than doing positive impact on like snake conservation, some people are involved in some uh, bad activities like illegal trading and other things. So those are not good in conservation. So in that way, now stringent guidelines are coming up from government, and those will be implemented soon. And lots of development has been made to keep track on those people, especially doing snake rescue and involved in illegal activities. But many, like we need also lots of volunteers, good people to help reduce the snake bite incidents in India. And so lots of work can be done in this aspect. And there is enormous scope to work on ecology and biology of species. Even in the common species, we don't have much knowledge about them. So like this, and few, like my dream is like a marriage. We need to see students like they should have some kind of naturalistic skills and taxonomy mixed together, so which help us in knowing the species in much detail. Rather than just describing a species, we should have enough knowledge about that particular species. For example, where they live and what are the microhabitat requirement, how many like eggs they lay, and how they live with other animals. So all these information, little bit of information combined together, it makes a wonderful contribution in the field of reptiles. So then do not shy away from using emerging techniques. So many people, they're afraid of doing molecular work. So if you are not familiar with molecular techniques, so you can collaborate with some good people, and then you can get your work done in collaboration. And you have to collaborate with lots of people, like to get some good thing, and people understand your contribution, and accordingly, like you get help from people. Then upgrade your skills constantly, disseminate them. Always you have to like be prompt enough to write papers, publish the things, whatever observations you are making. So now there are lots of journals which can publish your papers free of cost, and it can reach to a wide audience. So you can start writing, don't shy away. So you can also consult your teachers or your mentors to help you in writing papers. So in that way you can contribute, and your interest will be generated every time. And make taxonomy fun and relevant to society at a large, and whatever things you are doing, so let people know about it. And so all these things, like these are a small piece of advice from me, because means I have worked on different fields, different aspects, and finally, like we are in a stage like where we see we need our coming generation to be well equipped and smart enough to deal with the future crisis which we are going to see in near future. Maybe we have to save species, we have to think about our environment, and along with that, like we have to be good citizen to like save our nature from all these extinctions. Okay, thank you very much. If you have any questions, so I'll be happy to answer them. Hello.
Any
Yeah.
questions
Yeah.
from the audience? You can unmute yourself and ask, or else you can type the questions in the chat box. Any questions from the audience?
No questions. Oh, very nice. I think nobody could understand anything, or you understood everything.
Okay. Sir, I presume that's gone in detail. Bit by bit, you have enthused them into pursuing career in herpetology, provided in detail the methodologies that are starting from collection in the field and then on processing them and to go about identifying. Thank you for updating students, introducing them to the step-by-step process that is involved in taxonomy of reptiles. It was wonderful listening to you, sir, and students, I'm sure, are benefited by this informative talk of yours. And I believe there is a hand raised. BBZ43, go ahead, please.
Hello, sir.
Hi.
Sir, can you tell us about the scenario of snake milkers in our country?
Okay, so there are few only farms which are given permission to milk snakes for snake venom. So like one is Irula snake tribe, they are there, and they are catering the need for many of these anti-snake venom industries. Apart from that, there are a few small industries I have seen few in Uttar Pradesh. So those are like got permission from PCCF Wildlife to do small scale snake venom collection. So like that, and nowadays I think we need more of these snake milking farms, which can cater the need of the coming snake venom research, especially. So yeah, so like that, we don't have many more, but lots of illegal snake venom extraction is going on, like which are under the radar of forest department. But legally, there are very few. It is difficult to get a permission, of course, because when you are dealing with scheduled animals, so you have to be fair enough. You have to have clearance from PCCF Wildlife or Chief Wildlife Warden. So yes, Yogesh, it is difficult.
Thank you, sir. Another question from Prathamesh Joshi. I
Okay,
had
I
a
had a
doubt
doubt.
specific.
Uh-huh.
I had a doubt specific to sea snakes. How can we use locality data associated with museum specimens to map their distribution?
Okay. So Prathama is actually based on the specimen locality from where it has been collected. So that is the locality data for the particular specimen, and it shows that the species is distributed in that range. So in that way, it can be useful. So there is no problem like putting distribution of the species based on that museum specimen data. Okay.
Yes, sir. Thank you. Any more
Prathamesh, okay.
questions? Any more questions? Since there are no questions, I call upon Dr. John Paul to propose the vote of thanks.
Oh, somebody is asking.
Yeah, Prathamesh Joshi here.
Okay, hi Prathamesh. Hello.
Hello, am I audible?
Yes. Hello. Prathamesh, I can hear you. Okay, there is Ganesh. I don't know. Is there any funding from JDSI? Actually, JDSI doesn't provide funding as such to do any project. But if anybody is interested to join at JRF, so they have to, they have to join JDSI and to work with some scientists to work on specimens or field work or anything. So no direct funding, but if you join in any programs in JDSI, when like lots of advertisement comes for recruitment of JRF and all, so you can also write to director. Sometimes like you may get opportunities regarding doing some specific work on some animals. You can write to the director. You're free to write to any government body so that like if there is any vacancy, so they will also inform you.
Yes, Prathamesh, you can go ahead. You have raised your hand. Any more questions?
Yeah. So, what I wanted to ask is, when we look at localities associated with the museum specimen of sea snakes, they are like mentioning. So, for example, in BNHS, there are many specimens mentioning location as Bombay or Mumbai. So we don't know from where exactly that has been collected. Also, it might also have been collected from, offshore.
Okay.
So, is there any way to standardize that data or use it to map their distribution?
No, for sea snakes, basically, so if the species like if it doesn't come to coast or shore, so that means like the specimen has been collected from offshore. So that means that specimen is located or has been distributed in that particular area in the Bombay offshore. So if there is no, like in earlier collections, so basically people were depending on, or till now also, like we depend on fishermen's collection. Like if there is any trawling happening and they are getting the specimens from which, like if it has been collected, so it might have been mentioned from Bombay coast. Or if a researcher is going and collecting, so they also sometimes, like if they might give a particular geocode in it. So if it is not there, nothing to worry. But that particular species has been, or that particular specimen has been collected from the nearby locality of Bombay. So that's also fine. You can mention it as Bombay coast or something. There is nothing to worry much. It doesn't make much difference in uh, like in the distribution of that particular species of sea snake.
Oh,
okay.
Okay, sir.
Yeah.
Thank you.
Yeah. Okay. Thank you all.
Thanks.
Then, yeah, any queries also you can write to me anytime. So we'll be help. I'll help you in any way you want to do your research in the field of reptiles or any kind of like animal taxonomy, like in my field especially. So if anybody is interested, they can also write to me to work on any aspects. If you have any doubt, also you can anytime contact me. Thank you.
Thank you, sir, for your informative talk and also for your patience in taking the questions. I now call on Dr. John Paul to propose the vote of thanks to sir.
I believe Dr. John Paul has network issues. As I said, Dr. Pratyush, thank you for sparing your time to share this informative presentation on the collection, preservation, identification, and also the ecology of reptiles. You're right in emphasizing the need for collaboration and how knowledge can be shared in that regard in documenting the rich diversity that our country has. So thank you, sir. As you said, students will get in touch with you and also researchers who are part of this webinar. With your presence, we want to do something in that regard. Somewhere in May, when we organized the diversity distribution of Indian birds, a conference, we launched the Josephite Forum for Birders, a forum for the students to conduct activities on birding. Similarly, we are also starting a Herpes Club. So it's all done by students, and we are going to launch that in your presence. So I'll hand it over to the student coordinator, Mr. Rakshit. Rakshit, you can share and tell about it. It's a short event.
Okay.
Yes.
Hello. Yes.
Hi, Rakshit.
Hello, sir. Good evening, ladies and gentlemen, the distinguished keynote speaker, Dr. Pratyush, respected dignitaries, the presenters, the audience, and my dear fellow Josephites. On this occasion of the national webinar on herpetology, it is an honor to announce the launch of the Josephites Forum for Herping by the Natural Science Association here at the St. Joseph's College, Autonomous Bengaluru. The sole objective of the forum is to take up field trips to document the spatio-temporal species diversity of the amphibians and the reptiles, and to organize talks, seminars, competitions, and other events regarding the same on the subject of herpetology. I would like to thank our principal, Reverend Father Victor Lobo, the the college management, the faculty at the Department of Zoology at St. Joseph's for their support. And I thank wholeheartedly Dr. Jayshankar Him, the department from the Department of Zoology, the teacher coordinator at NSA for his encouragement and enthusiastic support, which has been very vital in setting up this forum as well as organizing this webinar. My dear friends, college mates, and fellow Josephites, this is a fantastic opportunity to begin and pursue your journey into the world of amateur herping and a lovely platform to participate in sharing of ideas, experiences, and talks regarding the same. I am personally looking forward to welcome you all at the forum. See you there very soon. Thank you, and I will hand over to Dr. Jayshankar.
Thank you, Rakshit, for the launch and also to convey the objective of the forum. Dr. Pratyush, thank you.
Yeah.
It happened
Thank you.
in your presence.
Okay. Thank you. I'm honored. Yeah. Thank you.
It's really an honor. Thank you, sir.
Okay.
Thank you for accepting our invitation.
Okay. Have a wonderful time. Bye-bye.
Bye,
okay.
sir.
Bye.
We now move on to continuing our oral presentation session. We have 3 pending oral presentations. With the online, yes, and the technical ups and downs, we tend to stretch a bit. So we'll now start on with the oral presentation that we're pending. I hand it over to the chairpersons, Dr. Ganesh and Professor Pratibha, to finish with the 3 more pending presentations of Vanlal, Gospel, Yajnapati. After that, we directly go into the poster presentation. In between, we also have a reel prepared by one of our students, Mr. Dhruv, about the common herpetofauna around our residence. So over to the chairpersons to finish the oral presentation. Thank you.
Thank you, sir. Moving on to the next set of oral presentation. The next oral presentation is from Vanlal Siamwai. Are you there? Vanlal Siamwai?
Yes, ma'am, I'm here.
Okay, fine. So the next presentation, over to you. You can share your screen.
Is my screen visible?
Yes. You can put it on the presentation mode.
Okay. Good evening, everyone. My name is Vanlal Siamwai, PhD scholar from the Department of Zoology.
Sorry, Vanlal.
City.
Sorry to interrupt. Can you put it on the presentation mode, please?
Wait a minute.
Yes.
Is my slide moving?
No, it's not.
Is it moving now?
I can see your presentation, but it's not moving. You need, yes, the slides are seen, but I need you to put it on the presentation mode, please. It's right below. On the left-hand corner of your screen, you have those buttons present next to the screen enlarged, and you can put it to the presentation mode.
The slide is not still moving, ma'am.
Oh, can you refresh it? Can you refresh it and start your slideshow again? Your screen is visible.
My screen is move as looking at from my friend's laptop.
Ma'am, my slide still cannot move. So
Just move your screen, just change your slides. Is it possible?
changing, but
slide change is possible. See right down on your left hand side corner, that is to your right hand side. You have this 83% present. That minus and plus sign you're seeing there. Right hand bottom corner.
ma'am, just give me a minute.
Sure.
Okay, shall we go with the next presenter, Mr. Yajnapati, and then come back to Vandlal with the chairpersons?
Yes, sir. Yes, sir. Not a problem.
If Mr. Yajnapati is there here, you can share, please.
Excuse me, sir. Is my screen being shared?
Not yet. I think Vandlal should stop sharing the screen. Vandlal CMV, can you please stop sharing your screen so that the other presenter can share the screen? Yes. Yajnapati's screen is
Okay,
visible. You can put, yes, it is visible. You can put it on the presentation mode.
Good evening, everyone. My name is Jack. I have pursued my master science degree in wildlife and biodiversity conservation from North Odisha University. My presentation topic is herpetofauna of Simhachalam Hills and Kambalakonda Wildlife Sanctuary. Is my voice audible?
Yes, your voice is audible, but can you put your slides on the presentation mode, please?
I have one actually. Is my screen being completely shared?
Yes, your screen is shared.
Full screen.
We can see your slides, but I want you to enlarge the slides so that it's completely visible. So I want you to put on the presentation mode. You know where exactly you have that 64% on the right hand side corner. Right next to that, you have this button.
I just
you
clicked
can.
it.
Okay.
Now is my screen being shared?
The screen is shared, the screen is visible, but I want it in the presentation mode. Yajnapati, please.
Yeah, it is in the presentation mode from my window, actually.
Okay, or else
You
Professor
continue
Pratibha,
with it.
yeah, let him continue. Thank you.
Continue with it. Fine.
Okay. Kambalakonda Wildlife Sanctuary and Simhachalam Hills are two separate study sites of my study area. The Eastern Ghats are being considered as a biogeographical zone on the basis of geomorphology and biodiversity point of view. Yet they are till date unsettled due to.
Yajnapati. Sorry for the interruption. Dr. Arun here.
Okay.
So actually, the screen is not properly visible. Okay,
Yeah.
if you can just go to the bottom of your what you call screen, there is 60%, and there is an what you call window-like option. You just click on that. Okay, and it will be in the presentation mode. Okay.
Yeah.
60% or some percentage is there towards the bottom corner. Okay, you have clicked to add slide, add notes, that bar is seen no at the bottom.
Yes.
Click to add notes. Okay, if you just go to the exact right corner, you have 64% percentage bar is there. Okay, next to the
Yes,
percentage bar, that the slide. Yeah, you just click on that at the bottom of your slide.
Actually, I just did, sir. Yeah, I just
Okay, what you can do is, okay, stop sharing now. Again, go for resharing and then do the same thing, sir. Because whatever you just what you call present it is audible, but the screen is not visible.
Yes, I just on it. Excuse me, sir. Is my screen like completely visible?
Your screen is visible, yes.
It's not in presentation mode. Okay, so I'll just give an this thing. Okay, instruction. You just follow that. Okay, in your slide there is an what you call circle with color, right? Circle. Okay, in your slide towards the right bottom.
Yeah.
Okay, you have a circle, right?
Yes.
In the design. Yeah. Okay, just below that. Yeah, just below that. Just below that. Okay, you have a small taskbar, right? Below that. Okay, in the screen. Taskbar. Yeah. So you just below that. Sir.
Yeah. Okay, sir.
Okay, and you have that percentage and window. Yeah, exactly where you have just there only. Exactly down, sir. Exactly down. Click on that, sir. Yeah. Click on that.
I did, sir.
Towards the right, you can just.
In my laptop, it is.
Okay, fine. Okay, you can just continue, sir. We'll see.
Okay.
Try doing a double
shall I
click.
continue with the presentation?
Yeah, you can. Try doing a double click and then you can continue.
Just it's still not working.
Then you can continue.
Okay, fine. My presentation is on herpetofauna of Simhachalam Hills and Malakunda Wildlife Sanctuary.
I believe the audio is not with us. We can't hear the speaker.
Yeah.
So can we go on to the next presenter? They can come back a little while later.
Yeah, yes, that's what I feel. Okay. Mr. Yajnapati, I believe there is some audio problem. You can stop sharing. We will let the other presenter, either Gospel or Vandlal, and then come back to you. Please accept her proposition. Okay,
Yes,
thank
sir.
you. Whenever there is an S, the audio is there, but unfortunately, the technicality, nothing in our hand. No problem. We will pursue it slowly. Yeah, is Vandlal or Gospel ready? Please go ahead. Both of you are from the same lab.
Yeah. Sir, Gospel is here.
Please proceed. Proceed. Share your slide. Chairpersons, please take note. The presenter is Gospel.
Yes, sir. Thank you.
Yes, Jay.
Ma'am, is it Is it coming? Is it my slide is coming?
Your slide is not coming. Yeah, I can see your screen.
Is my slide coming?
No, no, it's not there. You have to minimize this. Yes, right. Minimize this.
Yeah. Now is it visible?
The screen is visible, but I cannot see your windows. Gospel, you can stop share and share again.
Is it coming, Miss?
Yes, we can see. Can you kindly minimize your Windows team?
Yeah, now we can see.
Yes, you can minimize your teams. Yes.
Is my slide coming?
Yes, it is. Yes, yes. Good.
Okay.
Fine. You can continue.
Good evening, everyone. My name is Gospel Zoltan Moyamar, PhD scholar from Developmental Biology and Herpetology Lab, Department of Zoology, Mizoram University. My title for this presentation will be Confirmation on the Occurrence of Indian Flapshell Turtle, Lysamis punctata, and its distribution records in Mizoram, Northeast India. Indian flapshell turtle, Lysamis punctata, is a species of freshwater turtles belonging to the family Trionychidae of the order Trumdine. Currently, 3 subspecies, Lysamis punctata, Lysamis punctata andersoni, and Lysamis punctata vittata are recognized. Globally, the species has been reported from India, Pakistan, Nepal, Sri Lanka, Bangladesh, and Myanmar. In India, the animal is reported from Tamil Nadu, Goa, Kerala, Sikkim, Gujarat, Maharashtra, Himachal Pradesh, Jammu and Kashmir, Madhya Pradesh, Assam, and West Bengal. To date, 8 species of Trionychid turtles are known to occur in the country, like the Nilsonia gangeticus, Nilsonia lithi, Nilsonia hurum, Nilsonia nigricans, Tricha indica, Amida cartilaginea, Pelochelys, Cantori, and Lysamis punctata. Though knowledge about the distribution pattern and actual species of composition of Trionychid turtles in Mizoram is quite limited till date. However, 3 distinct species are currently known to occur, namely Lysamis punctata, Pelochelys, Cantori, Nilsoni hurum, and Amida cartilaginea. Although Matthew 2007 reported the presence of Lysamis punctata from Mizoram, but the knowledge about the species examination and the distribution of the turtles in the state Mizoram is not presented. So this presentation will provide solid information on the occurrence, confirmed occurrence and distribution patterns of Indian flapshell turtle Lysamis punctata from Mizoram. Here is materials and methods. Extensive survey on the Chelonian fauna of in Colosive district was conducted from September 2020 till date. We use direct sighting and visual encounter method, and then within the study site, reported occurrence of turtles and tortoises from the villagers are made use for effective field survey. Questionnaire along with photograph of Chelonian was also utilized among turtle hunters and fishermen. The vegetations of the present survey area, that is the Mamit and Colosive district, falls under tropical wet evergreen forest and semi-evergreen forest. And then the all of the collection sites which will I mention are of secondary forest type. And the annual rainfall of the study sites, Mamit district is around 2549.6 millimeter, and Colosive district is around 2,674.5 millimeter annually. And then species identification and sexing follows descriptive literature like Bupati et al. 2014, Das and Das 2017. Here are my results. Here is the representation of my results in the tabulation first one. We have the juvenile female, and the collection date is 17 October 2020, last year. And then the collection site is at Darlak village, Mami district. And here is the morphometric study. The carapace length measures around 8.4 centimeter, carapace width 8.3, and the plastron length is 8, and then the plastron width is 7.9. And the weight of the individual observed from this Darlak village is 0.0950 kilogram. And here is the coordinates, and it is 68 millimeter above sea level. And then the second one, adult female, we collected on 20 March 2021, and it's from Rahum, North Liman Kolasiv district. And here is the morphometric measurement. Carapace length 23, carapace width 16.3, plastron length 21, plastron width 21, and weight is around 1.240 kg. And then the third one is also a juvenile female. We collected on 15/5/2021, and it's from Zolnwam, Mami district. And here is the morphometric measurement. Carapace length 7.8, carapace width 8, plastron length 7.5, plastron width 6, and it weighed around 0.0715 kilogram. And the fourth one, we have adult male. We collected on 26 excuse me. So it's from Butangpai, Kolasiv district, and here is the morphometric measurement. The carapace length 23 centimeter, and the carapace width 16.5, and the plastron length 19.8, and plastron width 19.6. It weighs around 1.238 kilogram. And lastly, the juvenile female, we collected on 5th 2021. It's from Bairabi, Kolasiv district. Excuse me. Here is the morphometric measurement. Carapace length is 8.1 centimeter. Carapace width 8. Plastron length 7.8. Plastron width 7.5. And all the coordinates here are mentioned here. And then here we go to the figure. Figure 1 is the juvenile Lysamis punctata from Darlak, Mami district. And, the individual was at the time of collection, the individual was found buried in the mud at the bank of a pond. And here is the figure 2, adult female individual from Rahum Pond, North Liman, Kolasiv district. And here I have mentioned 3 different species of species. Mark A, B, and C. Here is A is a juvenile from Zolnwam district, and B, this one is from Butangpai, Kolasiv district, and C is from Bairabi, Kolasiv district. Collection site or the habitat. A, here is a fish pond at Darlak, Mami district. B is Rahum Pond, North Liman, Kolasiv district. C, a fish pond at Zolnwam, Mami district. And here figure 5. A is the pond from Butangpai village, Kolasiv district. B is a small stream at Bairabi, Kolasiv district. Is the updated distribution map of Lysamis punctata in Mizoram, Northeast India. So after identification and morphometric study was done, the specimens, all of the specimens that I encountered, we released back to their natural habitat. As we all know, Northeast India is considered the turtle hotspot, and also Mizoram is situated in the Indo-Myanmar Biodiversity Hotspot. So the biodiversity richness is quite high. So we hope for more positive and successful study of Chelonian from our state, Mizoram. And from this study, we also observed some turtles and tortoise are hunted for food and for zootherapeutic uses for treatment of some diseases like And so we suggest more public awareness for conservation of Trumdines and the biodiversity of Mizoram itself. So that's all is my presentation. Thank you.
Thank you, Gospel. Sir, Ganesh, sir.
Mr. Gospel?
Yes, sir.
Okay. Is there any conservation effort happening on these turtles?
Yes, yes, sir. Yes, sir. We are. We did some awareness to webinar, Google Meet among the higher students from different school, and then one student from the college, Serchip Government College. We did present awareness program, sir.
Have you done? Was there any species abundance study on this turtle?
Pardon, sir.
Have you done any species abundance work on this turtle? The number
Yes.
of turtles.
Yeah, we did some more studies on the family itself, like Nilsoni Hurum, which we published last year at Journal of Triton Texas. First report from Mizoram.
No, I'm talking about this turtle.
But this turtle.
Decimus punctata.
You mean like?
Have you done the species abundance study?
Yes, sir. It's still continuing, sir.
Oh, you're doing it?
Yeah. Till date, sir.
All right. Thank you.
Thank you, sir.
Thank you. Moving on to the next. Any questions from the audience? Any questions? Thank you, Gospel. Moving on to the next participant. Vanlai Siamvi, are you ready? Can you do the presentation?
Yes, ma'am, I'm ready.
Sure. Okay, continued.
Is my screen visible?
Yes, it is visible. Yes, it is in the presentation mode. You can continue.
Yeah. And good evening, everyone. My name is Vanel Siamoi, a PhD scholar from Department of Zoology, Mizoram University. And my topic is larval oral morphology of the white-lipped tree frog, Polypidage broweri from Mizoram, Northeast India. Ongoing to introduction, the oral discs of tadpoles are relatively conserved across different genera and families. However, the ecological as well as the dietary specializations of the tadpole has been shown to cause wide modifications in the oral apparatus, which is a reflection in their
Vanlal Siami, your voice is not audible.
She's changing slides, but voice is not audible.
Yes, sir.
She's mute.
Vanlal Siami.
Is it audible now?
Yes, yes, you're audible now. Thank you.
I'm going to continue from this slide.
Yes, sure. You can continue from this slide.
Polypidage broweri was first described by Vogt in 1911. And the genus Polypidates currently includes 25 nominal species and has a distribution ranging from eastern India, Southeast Asia, Philippines, and Borneo. And they are characterized by a relatively smaller size, the snout vein length ranging from 45 to 65 millimeter. And it was reported from Taiwan, tropical and subtropical China, Myanmar, Northeast India, and Guam. And Lan Mansang reported the occurrence of this frog in India from Mizoram, and they are inhabitants of natural as well as artificial ponds. Our present work report the first morphological structure of tadpoles of Polypidates broweri. And materials and methods. The studied specimens were collected from their natural habitat during the month of June to September 2021. The collected specimens were reared in laboratory conditions. Developmental stages of Polypidates were diagnosed based on Gusner, 1960. Description of oral disc and labeled tooth row formula follows the method provided by McDermott and Altic 1999 and Altic 2007. Different Gusner stages of tadpoles were fixed in 1:1 ratio of 70% alcohol and 4% formaldehyde and deposited in Departmental Museum of Zoology, Mizoram University, and the oral apparatus were studied under light microscope. The picture on the slide here are the natural habitats of tadpoles and the collected sites. And going through our results, the tadpole morphology. The body of tadpoles are oval shaped, snout slightly rounded, eyes lateral in position, nostril dorsal, nearer to snout than eyes, white dot present on the tip of snout. Tadpoles are grayish brown in color, covered with dark brown spots all over the body and tail. Abdomen and the fin parts are transparent. And the oral discs of Polypidates broweri are positioned anteroventrally. Marginal and submarginal papillae are present, and the keratoderm jaw starts developing at stage 25, and the labial papillae and big disc appearing by stage 42. The lower jaw set, here as we seen here, it is V-shaped structure, and the upper jaw has U-shaped arch with finely serrated edge, and the labial tooth row formula at stage 25 shows that there are three anterior tooth rows, A1, A2, and A3, and the first one is continuous, and there is medially interrupted from A2 and A3 anterior tooth 2 and 3, and the posterior labium P1, P2, and P3 are continuous. And between stage 26 and 34, there are two different labial tooth row formula, and at this stage, the mouth is more prominent, and the upper labium has 4 rows of labial teeth, A1, A2, A3, and A4, and they are interrupted medially from A2, A3, and A4, and there are 3 rows present on the lower labium, P1, P2, and P3, and in this case, the first row, P1, are sometimes interrupted and sometimes continuous depending upon the individual. And between stage 35 and 41, we also saw different labial tooth row formula. There are 5 labial tooth rows on the upper labium, A1, A2, A3, A4, and A5, and medially interrupted from 2nd to the 5th row, A2, A3, A4, and A5. And on the lower labium, there are 3 tooth rows. And the first tooth row, P1, are continuous or sometimes interrupted. And we also see another tooth row formula. There are 4 tooth rows formula on the anterior side, which is the same as the previous stages between stage 26 and 35. And on stage 42, The oral operata starts degenerating and gradually transforms into adult mouth. After attaining stage 46, the froglets feeds on carnivorous diet. And the present state represents the variations of label 2 draw formula at different developmental stages of polypidid species complex, and these differences are important for distinguishing the different species. On moving to discussion, comparing with the congenial species, including P. himalayensis, P. terrensis, and P. leucomystic, the oral apparatus of polypidid broweri show high variations, and the variation of labeled among species in particular reflects their microhabitat and the feeding habitat. These variations are a result of adaptive radiation of each species to explore different feeding needs within an ecosystem. As reported by various workers, the marginal and submarginal papillae play important roles in chemosensory and tactile receptors that controls the flow of water, attachment to substrates, shape of oral dish during feeding, and controls the ability to manipulate food and substrate particles. The strong upper and lower jaw sheaths serve as biting, scraping, and extorting structures. Although descriptions of tadpole characteristics are useful in taxonomy, the function of mouthparts, morphology of the labial teeth, and the functional significance of structural differences among species are still poorly known. Further studies need to be taken as the detailed descriptions on feeding biology and oral morphology of tadpoles of most underrun species is very limited. That will be all my presentation. Thank you.
Thank you very much. Over to Ganesh, sir.
1 minute. Hello, Vanlal.
Yes, sir.
Your studies seems to be very interesting, especially with regard to the larval oral morphology of this white-lipped tree frog. Apart from the differentiating species based on your study, you have given a wide account of the other aspects which can be explored based on your study. It was quite interesting. Since this is very new to me, I don't have much questions regarding this.
Thank you, sir.
Thank you very much for your presentation. By the way, did you have any idea about the feeding habit of this white-lipped frog, tree frog? Hello? Hello?
We are still observing on that.
Your voice is not clear.
According to their diet, we are still observing their diet content from the collected specimens in their natural habitat.
Okay. All right. Thank you very much.
Thank you, sir.
Okay, moving on to the next presenter. Yagnapati, are you there?
Yes, I'm online.
Okay, can you please start presenting?
Yes, ma'am. Just a moment.
Thank you.
Ma'am, is my screen completely being presented?
Yagnapati, your screen is visible, but it's not in the presentation mode. Can you reshare your screen again?
Yes, ma'am. I'm trying.
Okay.
Just give me a minute.
Yes, Yagnapati, your
Is my
screen is
screen
visible.
okay?
Yes, it's on the presentation mode. You can start your presentation.
Good evening, one and all. My name is Yagnapati Adadi from Visakhapatnam, Andhra Pradesh. I've pursued my Master of Science degree from North Odisha University from the Department of Wildlife and Biodiversity Conservation. Today, my presentation topic is Herpetofauna of Simhachalam Hills and Kambalakonda Wildlife Sanctuary, Visakhapatnam, Andhra Pradesh, Eastern Ghats. As Eastern Ghats are being considered as a biogeographical zone on the basis of geomorphology and biodiversity point of view. Yet till date they are unsettled because there is a huge lack of exploration and evaluation of the fragmented hill ranges. We know that Eastern Ghats start from northern Western Bengal and stretch till Tamil Nadu as a fragmented hill ranges. Kambalakonda Wildlife Sanctuary and Simhachalam Hill Range are also part of Eastern Ghats, and they're positioned northeast in the city with given coordinates 17 degrees 49 minutes north and 18 minutes east, and the both study sites are separated, or we can say they are there is a physical barrier of a huge road separating those two areas. Simhachalam Hills stretch about 16 square kilometers and are rich with medicinal herbs, grasses, shrubs, flowering plants, even some orchids as well, thus rendering its own microclimate. Whereas Kambalakonda Wildlife Sanctuary is composed of dry evergreen, tropical deciduous, and montane deciduous forests with a composition of Elianthus, Bahuinia, Chloroxylon, Cochlospermum, Ficus, Lana, Manilcara, Morinda, Tectona, and other species as well, along with some shrubland and meadows. Here there is a presentation of a map showing the boundaries of both Kambalakonda Wildlife Sanctuary and Simhachalam Hills. The top area which we can see is spread about 70 square kilometers. That represents Kambalakonda Wildlife Sanctuary, and to the southwest side is Simhachalam Hill Range. Elevation of Simhachalam Hill Range is about 370 meters to 527 meters, as the approachable elevation for any kind of study would be 369 meters, and the highest point would be 527 meters. Whereas Kambalakonda stands at just 198 meters. The city experiences basically seasonal variation in every 3 months. Thus, the monsoon is generally occurring from the month of June, with an annual rainfall of 990 to 1000 mm average annual rainfall, and temperature would range from 24 to 30 degrees. The forests of Kambalakonda Wildlife Sanctuary are basically tropical evergreen forests, shrub forests, subtropical and montane deciduous vegetations, along with grassy plains over the hilly terrain. These are common vegetation types of the study area. The herpetofauna is quite threatened by a notable cause of roadkills, hunting, and frequency of roadkills is very high during monsoon season. Since there is no prior research work done in both the study sites, a preliminary survey was performed from 15th of April to 15th of August 2021 to obtain a baseline data of herpetofauna with an authorized permit granted by the PCCF of Andhra Pradesh. Data collection. Various methods like direct sighting methods like visual encounter survey, road surveys, transact survey, litter survey during both day and night times were used for ground dwelling species. Flipping up rocks, logs, digging up soil, and and also pitfall traps were also used to encounter cryptozoic species and some endurance. Then following perennial streams to encounter frogs and some reptiles was also done. I also collected indirect evidences like mounting of snakes, auditory calls, and road-killed specimen examination, and also recorded them and photographed them. All animals were identified up to species level with the help of manuscript, field guide, etc. And also meristic details, morphometric analysis was also done for a few species, and some snakes and other reptiles which were caught in the field site are photographed and also released at the same time. Results of my survey included 38 species of herpetofauna distributed under 3 orders, 17 families, and 32 genera were recorded. 17 species of snakes belonging to 6 families, and 15 lizards belonging to 6 families, and 3 anurine families, namely Buphonidae, Rhacophoridae, and Dicoglosidae, along with 2 species of testudines belonging to 2 different families were observed. The reason for such a low amount of recordings for anurine species was just basically I sampled the study sites in pre-monsoon and basically at the end of summer, so I couldn't record much of the anurine species. Here's the inventory of herpetofauna recorded from Simhachalam Hills and Kambalakonda Wildlife Sanctuary. It includes 3 species that belong to Schedule 1 of Wildlife Protection Act, and 14 species that are categorized under Schedule 4 of Wildlife Protection Act 1972, and 5 species belonging to Schedule 2 of Wildlife Protection Act. Notable findings like Indian star tortoise, which is endangered, golden gecko, Calodactylus aureus, and there are also like a few Hemiphyllodactylus species as well in the Simhachalam Hills. And one more interesting thing I could definitely note was the occurrence of Oligodon teneolatus, that is a striated kukri snake. It was only encountered during monsoonal seasons in the foothills of Simhachalam Hills because it's basically the breeding season of Oligodon species. So a total of 38 species were recorded from the survey period. As I've already said that road kills is a major threat in the entire study site. You can see Eublepharis hardwicki is like a road kill, and then Ahetula oxyrhyncha. Not only these two, there is also sand boa, Eryx conicus. There is Python molurus and Russell's viper, Daboia russeli. They're also recorded as road kills. Basically, for all the factors responsible for the death of herpetofaunal community, road kills contribute to the most percentage. Speed limiting boards must be set up by the traffic authority in animal areas and sensitive zones, animal crossing areas and sensitive zones. There is a need of awareness among local inhabitants of people living in the vicinity of the protected area. There is a local community which is also an indigenous tribe called the Mannamdora tribe of Kambalakonda Wildlife Sanctuary. Basically, they are being relocated or being highly encouraged to get other lifestyles other than depending upon the forest. So there is also a threat to losing of indigenous knowledge and indigenous diversity. In the era of digitalization, there is also a need to rethink about mythical beliefs and age-old practices. While I have interviewed the people of the Mannamdara tribes, they believed in various types of myths and all. That is also some point to be a concern. Active snake rescue units must be informed in order to prevent conflict with human beings that may lead to fatality of either one of individuals. So there is also a need to establish snake rescue operations, which would lead to low human wildlife conflict. Uh, this is Indian golden gecko, Calodactylus aureus. Many nesting sites were also identified in both the study sites of Simhachalam Hills and as well as Kammalakonda Wildlife Sanctuary. There is also Certodactylus nebulosus, Indian clouded gecko, which were mostly nocturnal and which are only encountered during nocturnal surveys. This is another morph of Indian clouded geckoella. This is Hemidactylus CF Sushil Dattai. It is not confirmed as Sushil Dattai, but it can be identified as a Sushil Dattai because of a type locality. So this was a very first record from Kambalakonda Wildlife Sanctuary as of now.
Are you changing your slide?
Yes, sir.
We are unable to see the changed slide.
Yeah.
Okay.
Yeah.
May I know at which slide you are presently at, sir?
Yeah, Datta's Mahendagiri.
Yeah. Datta's Mahendragiri gecko. Yeah. Indian chameleon Zelynicus. It is one of endangered species now. There are also lots of Hemiphyllodactylus species. I couldn't identify a few that I have encountered. Bamboo pit viper, Craspidocephalus gramineus. As Pratyush sir has given his talk before, given his lecture before, he said that Vishakhapatnam is highly diverse with serpent of Hona, and it is also type locality for many species. One such is bamboo pit viper. There is also Typhlops brahmineus, the Brahmini blind snake. There is also Sushil Dattai, Hemidactylus Sushil Dattai, Datta's Mahendragiri's gecko. Its type locality is basically Simhachalam hills, and during my survey period, I could encounter one from Kammalakonda Wildlife Sanctuary. There is also Coleognathus helena, which has its type locality from Vishakhapatnam city. There is also Indian common crate, Bungarus caroleus, which also holds a type locality as Vishakhapatnam. This is an Indian star tortoise, Geochilon elegans. We can see it is dwelling in its habitat. That would be the end of my presentation, and I would like to thank Dr. Kuku Mahapatra ma'am, Assistant Professor, Department of Zoology, North Odisha University, for assistance with lab facilities during my morphometric analysis and meristic data collection and all. That would be it. Thank you.
Thank you, Agnapati. Over to Ganesh sir for your comments and questions.
Thank you, ma'am. As you said, it is the first time that you are studying on the herpetofauna of Simachalam Hills and Kambalakunda Wildlife Sanctuary.
Yes, sir.
Uh, it's a very good initiative from your side.
Yes,
But
sir.
initially, your description regarding the study site, both the study site, were quite elaborate. I think it went up to 10 minutes. Anyway, that's not a
Uh,
matter. You did between 15th April and 15th August, right?
yes, sir.
It was for about roughly for about 6 months.
Yes, sir.
Was this a monsoon
Uh,
period?
actually, sir, there were some COVID restrictions for which I couldn't sample the area in the month of mid June to May to June, sir. So that's
Okay.
again
Is this a
not able to.
monsoon period? Is it a monsoon
Uh,
time?
no, sir. Actually, the Kammalakonda Wildlife Sanctuary basically gets monsoon during early October, and June July is like we have slight showers during the that time of year.
Okay. Okay. You reported about finding about 38 species of herpetofauna. From
Yes,
is it
and
from both this study site?
yes, sir, from both the study sites, and
Okay. What?
few species were like not in both the study sites.
Uh,
It's just collaborative data, preliminary data for a baseline survey. Yeah.
Okay. What I feel is since the area is quite vast, and if you extend the time of your study, you may get more number of species. That's what I
Yes, sir.
feel.
Indeed. Yeah, yeah. Due to lack of I have surveyed the study site early monsoon, so I couldn't get most of the annurin species.
Exactly.
Yeah, and I only took recordings of the species I've cited directly. I could have also minervarious species vocal calls, but I haven't considered it. Some sampling would definitely bring some more species to the list.
Okay. Thank you very much. It was quite interesting.
Yes, sir.
Thank you, sir. Any questions from the audience? Questions? Okay, it's Vanlal CMV here. Vanlal CMV, are you here?
Yeah, she finished, Pratibha.
Sir, she has a question, sir. It's on the chat box.
Okay.
She is not available at the moment.
There is a question for her. The question is there an approach on the scanning electron microscope on magnification?
Okay.
She's not available to give answer. Sorry.
Okay, fine. If she is available after some time, can you ask her to please put it up on the chat box?
Okay.
Thank you so much. Okay, we now have completed all the oral presentations for the national webinar. My heartfelt gratitude to Ganesh sir for all his cooperation and his help. And I now hand over the session to Dr. Jaishankar sir.
Thank you, Pratibha ma'am. Thank you, both of you, the chairpersons, Dr. Ganesh from Christ University, and also Professor Pratibha from the Department of Zoology, St. Joseph's College, for running this show. My apologies for making you wait and extend and divide the session into two. Sorry for that, and thank you for bearing up with that.
Thank you.
Yes, sir. Thank you, sir. Thanks a lot.
Thanks for giving us the opportunity.
Welcome, both of you. With that, we are done with the oral presentations. We are overshot in the sense the online platforms are a boon, very great gift by technology. But along with that gift comes a technical glitches. I think with the good, we have to bear the shortcoming also. And also, this is going to be another 1 hour extension of yesterday. My humble request is, let us encourage the youngsters, the researchers. That is one way we propagate knowledge and also allow it to be shared among everybody. That's the reason we were not strict about 5+2 minutes and other kind of rules and regulation. We gave full-fledged timing for the young researchers to make their presentations. At this time, I'd like to thank all the chairpersons who have done the 3 sessions, yesterday 2 and today 1. My special thanks to Dr. H.T. Lalram Sangha from the Mizoram University. His lab has contributed 8 of the oral presentations of the total 17 that we received. Thank you, sir, for your immense contribution. We wish to associate with you in future as well. In addition to this, we have 20 poster presentations, which we will start now. Students are here. Please make sure that it is only 5 minutes because it's a review work that you're presenting. And these info poster uh, provision is made to encourage all the undergraduate students to expose them to the platform of presenting. That's the main intention, though it is not a hardcore original research work, but a review. Even to prepare that, this is how we should tutor and provide opportunity to our students, and they have done wonderful job in preparing the abstracts. Let's wait to see that. We launched the Josephite Forum for Herpes. The first initiative in that line is going to be a presentation, a reel on the common herpetofauna in and around our residences. Over to Dhruv, a student in St. Joseph's College, who will do the first opening documentary to the Josephite Forum of Herpes, for Herpes. Dhruv, over to you to share.
Yes, good evening, everybody. Yes, is my screen visible?
Yes, Dhruv, go ahead.
Yeah, so first of all, I would like to thank Jai Shankar sir for giving me this opportunity to present right forum. I'm very honored. And I would also like to thank all of the participants for their very insightful presentations. I've learned so much from all of them. My topic that I will present today is the collage of common herpetofauna around us, Bangalore city. So from the oral presentations, we've seen that herpetofauna, especially in India, is very diverse. We have so many different species that can be found in a variety of ranges. And while it is astounding to see that, it is always important to also see the herpetofauna we have in our residential areas, study them. As Pratyush sir also mentioned in his talk, herpetology is studied very extensively and taxonomy is done, but there's so little we understand about the animal behavior, and so much more studies can be done into this. For the common species we already know. So my presentation today is going to center around common identifiable herpetofauna that I have observed myself. And it's also a sort of introduction into any students or anybody really who wants to get into herping. So yeah, with that, I shall begin. So first, we look at some lizard species. So the first species I'd like to share with you all today is the Mysore day gecko, or Snemaspis mysoriensis. So this is a very, very common, possibly the most common gecko species you can find, ranging from Bangalore to Mysore. And even though the species is so common and so well distributed, there is very little studies that has really been done on its behavior, on its ecological role, and other aspects. So they are a diurnal species. They are a member of the day gecko genus, or in Snemaspis, which is a very diverse genera of day geckos ranging on Asia and Africa. They lay 20 to 30 eggs in communal oviposition sites. So that is the reason they are able to adapt so well to urban situations. This is because if you, for example, have observed walls, stone walls, crevices are present, a communal oviposition site can be made. Even the encrusting of regular concrete walls provide a place where they can lay their eggs. So they can be identified by their spotted patterns, rounded pupil, flattened body, and um, the slender clawed digits. So that is about the Mysore day gecko. Next, we have the common house gecko, or it's even known as the Asian house gecko, Hemidactylus frenatus. This is again a very widespread species of gecko that's found all across the Southeast Asian countries, and in some cases this can be an invasive species. So it is a slightly larger size, a medium-sized gecko species, I would say, and it competes with parthenogenetic species of other geckos. So they do not tolerate other species of lizards that well, and if this species encounters another species of lizard, it will compete with them directly for food resources and others. So the way it acts as an invasive species is if there is a parthenogenetic species of lizard present in the same area as this lizard, parthenogenetic means that most or all of the members of that lizard species will be females. The relatively aggressive male species of this lizard will compete with them directly, and they can displace species of lizards. In Bangalore, though, I have not come across any documentation of this species really being um, invasive as such, and it can be found around most urban areas. It is a synanthropic species. It is adapted to live in urban conditions with higher light, and it's versatile. It hunts a wide variety of invertebrate species. So the Rockagama, or more specifically, the peninsular Rockagama. So the Rockagamas can be differentiated into the peninsular Rockagama and the eastern Rockagama. And in the Western Ghats, the Karnataka side, we have the peninsular Rockagama, Samophilus dorsalis. Samophilus literally translates to sand-loving. Um, the name may be slightly deceptive because this species tends to live on rock or rocky outcrops. So, and this habitat is found throughout the Dharwar Craton and even places in Bangalore. So these species have even been encountered in our college, St. Joseph's College, I think once by Professor Wilson. They can also be found on Lalbagh Rock, but that is in the areas which aren't very crowded with people. So the males, there is a sexual dimorphism in this species. The females tend to have a slightly more cryptic coloration with white dots and to help them camouflage with the rocky outcrop. And the males are a darker shade, as seen here, with a bright red, a very vivid bright red or yellow head with a small streak across the eye. So to identify these, to come across these species, if you were to say trekking on a rocky outcrop or were in Lalbagh Rock, is difficult unless you get very close to them because of the cryptic camouflage and their behavior. They tend to stay still whenever they feel threatened and lie down on the rock surface. You wouldn't know right unless you get right up to them and they run away. So that is about the Rockagama. And next we have the Oriental Garden Lizard. So this is again another it's a common species of lizard that can be found all throughout Bangalore, but they tend to stay away from the urbanized areas and usually can be found around garden areas. So another interesting name that I came across of this lizard is bloodsucker. So it is known as bloodsucker in local languages in some areas and even in English. This is due to the fact that males during the breeding seasons develop a very bright red colored head, and it looks almost like it's covered in blood. So again, a fairly common and this is a larger species of lizard. It can grow longer than 10 centimeters, and it is an agamid. So that is about the lizard species you can find in Bangalore. Next, we have snakes. Now, before I continue, I should mention that for the snakes centered around Bangalore, there are a wide variety of species, but I've limited them to only 3 that can be found even within the residential areas at times. So first, we have the rat snake, Pythias mucosa. This species of snake can be found sometimes around garden areas. They can migrate and be found within garden areas. And they are a diurnal species of snake. They tend to hunt during the day, and they are non-venomous, of course, so they really don't pose that big of a threat to people, especially if they're found in garden areas. So whenever rescue calls happen and rescuers are able to identify that it's a rat snake and it is found in a garden area, they tend to leave them alone. This is because, for starters, they are non-venomous, and also they're very important for the ecosystem. They control the rat population, populations of small lizards, as we discussed, the common house gecko is an invasive species. So this directly controls the population of them. So it is an important species of snake. It can be found in within garden areas. Next, we have the checkered keelback, also known as the Asian water snake. So, as the name suggests, the species can be found in and around water bodies, usually, and they can be identified by their check-like patterns on their body. It looks almost like a checkboard. Keelback refers to the shape of their back. It is shaped like a keel of a ship, the bottom part of a ship. It's slightly raised ridge. And yeah, this is also a non-venomous species of snake, although its saliva does contain some anticoagulant properties. So if bitten by one, the wound doesn't tend to clot. This is kind of common with water snakes. And next, we also have the spectacle cobra. This is a very widespread species of cobra in India and especially in Karnataka, and can be found in a wide variety of places. And rescue calls do even happen from residential areas. So, as its name suggests, it can be identified by a distinctive speckled pattern on the backside of its hood. It is a venomous snake. It possesses a neurotoxin that affects the synapses of the neurons, which eventually leads to paralysis. So these three species of snakes are very important because they are all very similar and they can be found throughout Bangalore. And due to the range of coloration from light yellow to darkish black, as seen in this first photo, they can be very easily misidentified. So if someone is herping and is looking for snakes, you should take care to identify that the snake you're observing is not a venomous cobra, because that would be bad. And also, the checkered keelback and Indian rat snakes can be Batesian mimics of the spectacle cobra. They try to puff up the neck region and hiss aggressively to try and mimic a spectacle cobra when threatened. So this is another thing that can be noted. And yeah, so next we have the amphibian species. So as we've seen so far in these oral presentations, especially by the first one, diversity of amphibians in India is very great. The first speaker today, Mr. Deepak, who was talking about the diversity of amphibians, mentioned that a species of frog has been identified in Bangalore as recently as 2020. This is just a testament to how common amphibian species can be. So I decided to narrow down my amphibians to 3 common identifiable species that can be found in Bangalore, especially if hopping during the night. So the first species is the Asian common toad. So this is again a very, very common species of toad that can be found all across Southeast Asia, and they can be an invasive species. They are an invasive species in some of the island chains in Papua New Guinea and Oceania. And in Australia, they are actually a bit of a problem as an invasive species because they compete with the local cane toad populations. So the easiest way to identify the Asian common toad is the crest like structure that's present above its eyes. It's a hard crest, and even the enlarged parotid glands, although that is very secondary. So yeah, this is a very common species of toad, and they tend to breed around shallow lentic water bodies where they produce their eggs. And during the night, especially during the rainy season, they are very common and very easy to encounter. Even in the college campus, they've been found quite a bit. Next, we have the Sri Lankan painted frog. So this is also again a common species of frog, Operodon taprobanicus. So the identifying feature of this frog is the reddish patterns that can be found on the top of it. It is again a smallish sized frog and usually can be found in the night, and they tend to stay around probably under rocks and in within crevices. So they are not so easy to find if you are only going for a night walk. You'd have to do some digging around. This species has also been encountered at a college campus. I think by one of the previous speakers the other day, Saklain. he mentioned that he was able to document the species within the college campus, and it is fairly common around Bangalore to see. The next species of frog we have is the termite nest frog. So again, it's identifiable by the spots on its back. It tends to be a smaller species of frog that is pretty widespread around Bangalore also. If you are going out to look for it, the most common spots to find it, as its name suggests, is around termite mounds and even under rock crevices. But especially during the monsoon time, these frogs tend to come out. So you can see them even in indoor regions also. One interesting thing I came across with these frogs is small species of frogs tend sometimes have a mutualistic relationship with invertebrates like tarantulas and scorpions, species that can easily kill them. So sometimes you can find these species of frogs in the same microhabitat as a scorpion species or as a tarantula. So yeah, with that, I end my presentation. Thank you for your time. And if you have any questions,
Yeah, if the audience or students have any comments, clarification to share, we can do it fast and then move on to the poster presentations. Okay, if there aren't any comments, we are now. Thank you, Dhruv, for that launching event of the Josephite Forum for Herping. Okay, thank you. We now move on to the last segment of the 2 days national webinar on herpetology. This is the info poster presentation by faculty and students. These are not original research, but a review that the presenters have made. Let us encourage them, provide a platform for them to present their skills. This is divided into 4 sessions. Each of them have 5 presenters, and every presenter gets around 5 minutes, 1 or 2 questions by the chairpersons, so that we can manage it finishing by 9 o'clock today. So we are now moving into the first session. May I request if the chairpersons are around? Mr. Lal Mohan Sangha is here.
Yes, sir.
Thank you, Lal, for being there. Dr. Manjula? Yeah. I think Dr. Manjula has just joined us. Dr. Manjula, can you hear us?
Yes, Dr. Jay, I can hear you.
Yeah, thank you. You have rightly come at the right time. We are starting now, the poster presentation.
Yeah, thank you.
Yeah. So over to you, Dr. Manjula and Mr. Lal Mohan Sangha. Lal Mohan Sangha made this wonderful presentation yesterday. He's from the lab of Dr. H.T. Lalrim Sangha from the Mizoram University, a lab that is vivid in activity. You can see all the 8 presentations happening and all 8 of them with their guide working together. Now that was a cohesive unit. We really love to see such labs and co-workers. So over to the chairpersons. We have 5 presenters. Please confirm Hemalata, Chaitanya, Joshika, Annie Maria, and Malavika. Can you confirm your presence, Hemalata?
Yes, sir, present. I'm here.
Thank you. Chaitanya?
Yes, sir, present.
Joshika? Joshika is not here. Okay. Annie Maria?
Sir, Ranjita will be presenting, sir.
Ranjita, good. Okay. Malavika?
Excuse me. Joshika is present, sorry.
Joshika is present. Malavika?
Yes, sir, I'm here.
Yeah. Okay, done. We'll start with the presentations. Over to the chairpersons. Chairpersons also 1 or 2 questions. These are review. We will try to manage time. Students, please keep in mind the time limit. Over to the chairpersons. Thank you. Dr. Manjula is from Reva University, sorry.
Yeah.
And Mr. Lal Mohan Sangha from the Mizoram University. Thank you for accepting our invitation.
Yeah, thank you. Thank you, Dr. Jay. And all the participants, all the best. So first,
वो लोग आगे हैं, हम पीछे हैं, वो लोग इतना
Please turn off your mic. Your personal conversation is made public. Thank you.
Okay. So Jai, can we start with Hemalata, right?
Yes, ma'am. Yeah, please.
Yeah. Yes, Hemalata, you can go ahead.
Okay. I'm Hemalata. So I'm assistant professor working in NM Carey Degree College for Women's. So today my topic for the poster presentation is camouflage in reptiles. Okay, I'll just present my poster. Is it visible, madam? Madam and sir?
Yes, it's visible.
Yes, I think you have started sharing. Yes, now it is visible.
Okay, fine. Okay, so I'll be starting the topic. So good evening to all of you who have, thank you for considering for the poster presentation. So the topic for the discussion is the camouflage in reptiles. So all of you know that the reptiles are cold-blooded animals, have scales on their body, and usually they breathe through the lungs, lay eggs. So these are the common general characteristics of the reptiles, and they are so they are observed worldwide. So they are cosmopolitan in nature. So as they are spread all over, yes. Hello. Am I audible?
Yes, madam. Proceed, please.
Okay.
Yes, ma'am. Please go ahead.
Okay, so they exhibit variation as well as the adaptive mechanism. So this is the camouflage is nothing but they show an adaptive mechanism, that is the defensive mechanism, where they itself disguise themselves to the surrounding. So they're disguising in a sense to hide from the predators. So they do color change. So the color changing, the pattern, so it shows different colors, so the patterns to escape from the predators. So they match with the surroundings. So the matching can be like most of the lizards. So the example, the most familiar one, what we all know is that the chameleon. So chameleon so changes the color within a minute or a second, which resembles the surrounding. So that is the best example. And even the snakes for hiding from the predators, they do mimic another snakes, which are like venomous snakes. So that is the one. So those are the like camouflaging is widely seen in the reptiles. Even some of the amphibians also, like highland, all they show mimicry. So as we are focusing on the reptiles, so this is the one. So the camouflage, mainly the animal masking the location, identity, and the movement. So as for example, if you take the example of a leaf-tailed gecko, so they actually resemble a leaf, almost a leaf. I have put a picture in the poster. You can all have a look on that. So leaf-tailed gecko. So the movement is also like that. If the leaves moves, the animal also moves like that. So what are the things which are responsible for this coloration? So the coloration is mainly due to the presence of melanophores. So these melanophores are present in the dermis of the skin. So the melanophores, whenever the animal receives the signals through the eyes, wherever the predator is there, so they receive the signal and sends to its cognitive nervous system. So there is a combination, there is a physiological combination of both hormonal responses as well as the neuronal responses. So most of the things like I have told you that melanophores are the pigment cells which are involved in the color changes. So these melanophores are activated when the sensation of the predator is received, and they start synthesizing some of the colors. So the melanophores usually they secrete melanin, which is responsible for giving black or brown color to the animal. And some of the other chromatophores are there, like depending on the colors. Like if it is erythrocyte, it gives the red color. If it is xanthophore, it gives the yellow color. If it is leukocytes, like erythrocytes and wheel cell, no, like erythrocytes, leukocytes, they are giving the red blood cells. Same way, here also the leucophores are responsible for giving the white color, and the cyanophores gives the blue color. So the colorations all depends on the this neurological as well as the hormonal cells, which activates the pigmental cells for secretion of different color. Okay, so that is the one. And usually the neuronal responses are much quicker. So most of the chameleons, instead of going to the hormonal responses, they are, as they change the color in minutes, second, so they usually depend on the neuronal responses. So the main, if a hormone is involved, so the main hormone involved is that, so the main hormone responsible is the melanocyte stimulating hormone, MSH, that activates the melanophores. So coming on to the, so this was the physiological part as well as the hormonal response and what are the cells involved in the color changes. So next is that we have got different types of camouflages. So I have named a few of them. So first one is the cryptic coloration. So cryptic coloration is nothing but that makes the animal difficult to distinguish against their background. So the best example is with which I put in the poster is that leaf-tailed gecko, then both they are the gecko itself, Santanic leaf-tailed gecko, giant leaf-tailed gecko, and some of the sidewinder rattlesnakes also shows the cryptic type of coloration. And next type of coloration is camouflage is that dazzle camouflage. So you might have heard like dazzle camouflage. Best example can be seen in the animal zebra. So the same way here also the reptiles also show the pattern. It is almost like creating illusions. So it is a pattern designed to form illusions. So where they actually create illusion so that the prey doesn't distinguish between the surrounding, so that they have blended like that. It creates the illusion. So it mainly depends on the patterns, as well as the patterns can be like stripes pattern, zigzag patterns, and it usually reduces the predation most of the cases. So examples for this dazzle camouflage is that panther chameleon, chippy house guest. So all these are the examples for the dazzle camouflage. And next coloration, the type of camouflage you can see is that aposematic coloration. So these are the warning coloration used by the harmful organisms, like most of the cases poisonous snakes, for example. So these are the warning colors. So by seeing the coloration itself, it gives the signals to the predators, potential predators, that they recognize the danger. So that's what, when a warning is given, like when the predator sees that, so there is a danger, we should not go there. So that type of warning coloration is called as aposematic coloration. So this is used usually by the harmful organism, snakes, that is venomous snakes usually. So the best example is the coral snake. And so that is about the aposematic coloration. And the next type of camouflage is the mimicry. So they do mimicking. So the mimicking can be in the form of voice, but here or mimicking somebody else. So the mimicry here is called as Batesian mimicry, where the animals mimic the coloration of the poisonous animal. So a non-venomous snake can mimic like a venomous snake, so that the predators will think that, oh, this non-venomous snake might be a venomous snake. For example, I have put in the poster the non-venomous snake, like example is wolf snake, that mimics the venomous venomous snake, that is the Malayan Cryate. Example is Malayan Cryate. So that was about the Batesian mimicry. And next, last one is the caudal luring. Okay, so this is the caudal luring. You can see where the tail of the snake is waved. So waving of the tail like a small worm, so that can attract the prey. So you can see in the picture, so where the snake tail is almost like a worm, small worm, and it waves. So these are the few types of there are different types of other different types of camouflaging properties or types of camouflages are also there, but I have briefed it about the what do you mean by the camouflage, what and all the physiological part involved, and a few types of camouflage along with the examples. So thank you.
Okay,
Yeah, very good. Yeah, please go ahead, sir.
Okay, ma'am, I have one question.
Yes, sir.
In terms of camouflage, let's keep camouflage on the other side, and I want to ask, do melanophore has any other significance to reptiles? Melanophores.
Melanophores actually, along with the iridiophores, they might be reflecting the lights also. They in combination with the iridiophores, they reflects the light and a particular color.
Okay, thank you very much. And I would also like to say I add a small account that melanophores in king cobras.
Yes,
Let me add a small account. Melanophores in cobras. Melanophore is a pigment which is present on the skin, on the outer surface of the skin, and when documenting the snakes of Western Ghats, it was found that king cobras use melanophores to absorb light from the sunlight because it absorb heat from sunlight, because black coloration doesn't reflect much light than white coloration. That means if they can change their color to black using the melanophores, that means it will increasingly help in their digestion during basking. Okay, thank you very much for the presentation.
Okay, so thank you very much.
Yeah, Hemalata ma'am, very good presentation, and even the poster you have done it very beautifully. It is very good. Yeah, I just have one question for you.
Yes,
you
ma'am.
spoke about camouflage in all the reptiles. Have you seen any kind of such activity in the aquatic animals?
Aquatic, I have not gone through. Aquatic animals, general animals, like amphibians, like I have heard of hyla and all. Aquatic, I'm not aware.
Okay, fine, ma'am. So actually, it is even cuttlefish, if you have seen, no? It changes its texture itself.
Okay.
Even octopus is known for that. Okay, fine, ma'am. It was a good presentation. All the best.
Thank you, madam. Okay.
So, Dr. Jai, can we go ahead with the next presentation?
Yeah.
Or any other questions?
No, restrict it. You both only can ask and make it 5 minutes. Yeah, thank you.
Yeah, so I think the second presentation is by Chaitanya Ora. Chaitanya
yes,
is here.
Yes, good evening, ma'am.
Yeah. Hemalata ma'am, can you just stop sharing your screen? Yeah, Chaitanya, you can start.
Yes, ma'am. Ma'am, is it visible?
Yes, it is visible. Go ahead, all the best.
So my presentation is an abstract on Sicilians of Western Ghats. What is a Sicilian? Is it a worm or is it a snake? It's none of those. Sicilian is actually a legless amphibian. It falls under one of the three modern orders of amphibians, the Apoda, which it belongs to, along with Anura, the frogs, and Urodela, the salamanders. Not much is known about these unique creatures, as most of them are fossorial or subterranean, so finding them becomes a difficult task. Sicilian is a term derived from the Latin word meaning the blind ones, but they actually have small eyes which has limited dark light perception, which they use to catch their prey. In some species, the eyes are covered completely by the skin as they are fossorial, so they have lost their eyesight. Their anatomy is highly adapted for burrowing purposes. Sicilians actually have a backbone and a skull. They use their strong skull and muscle to drive through the dirt like a piston in a car engine. Caecilians also have tendons which help them in burrowing. Unlike the snakes which have scales on outside of the body, Caecilians have scale inside the skins. They have smooth skin which is covered with toxic mucus secreted by their skin. They also have a pair of tentacles for olfactory capability. Caecilians also possess needle-like teeth which they feed on insects and other vertebrates. Though their diet is not well known, ongoing studies suggest that they may have venomous saliva, but nothing has yet been proven. The most fascinating thing about Caecilian is their reproduction and parental care. Different species have different way of reproduction and parental care. Some are oviparous, laying eggs in water and or moist soil, and some have evolved to take care of live young ones. They are viviparous. Some species of Caecilians have a unique, bizarre approach when it comes to feeding their young ones. The young ones feed on the skin of the mother. That's right, they eat their mother's skin. This is known as maternal dermatophagy. It's actually a special layer of skin twice as thick from the normal skin designed for this purpose itself by a brooding female. The skin cells are said to be special in this layer as they provide the young ones with more protein and fat. The skin is scraped off by the young Caecilians who come equipped with a set of temporary teeth. The mother is uninjured in the process. If you think this was bizarre enough, in viviparous species of Caecilians, the live young ones start to feed on the swollen lining of their mother's oviduct before they are born. These are generally the Kenyan species of Caecilians. Both the cases fall under matrophagy, extreme form of parental care. All species around the world are found in the tropical environments. In India, they are found in the northeastern region, and a huge diversity is found in Western Ghats. As all of us know, Western Ghats is one of the 8 hotspots of biological diversity of the world. Western Ghats harbors a huge diversity of amphibians, and 80% of them is endemic to the place. Caecilians of Western Ghats are no exception to this. Two families of Caecilians, the family.
Sorry to interrupt, but can you move on to your results as we have a time limit here on the poster presentation. Please, can you move on to your results?
Sir, it's just the extract is over actually.
Okay, then proceed.
The two families which are Indotyphlidae, which contains the two genus which are Indotyphlis and Genophias, and other family Ichthyophidae contains two genus found in Western Ghats, namely Ichthyophias and genus Ureotyphlis. There are nearly 26 species of Caecilians found in Western Ghats. Most data regarding their biology and conservation status are deficient. Most amphibian species are declining in the world, so we need to know more about the unique creatures and conserve them. Thank you.
Okay, Chaitanya, here I have few suggestions.
Yes, sir.
You could have made it a little pictorial presentation, no? So that it will be easy for you also.
Yes, ma'am, but actually I thought that in the abstracts there should be no pictures. So.
Okay, fine, Chaitanya. Chaitanya, can you tell me what kind of defense mechanism these Caecilians have?
Ma'am, they use the they secrete the toxic in their mucus, and that's all over their skin. So that's their defense mechanism.
Okay, fine, Chaitanya. All the best. Sir, you have any question?
Okay, I have one question. Chaitanya, are there any keys on how to differentiate between how do they differentiate identify different species of Sicilians?
Sir, I think that by I don't know much about it, but it may be by the genetics.
Okay.
Collection of
Okay, that is very true because now the classical taxonomy is a little bit hard as molecular analysis has come out. It's actually the annuli count, the number of stripes are counted in most of the cases. Okay, thank you very much. Nice presentation.
You,
Okay, sir, then we'll go ahead with the next presentation. Joshika?
Yes, ma'am. Good evening, everyone. I'll be sharing my screen. Can you all see my screen?
I'm not able to see your screen, Joshika. Are you sharing?
Yes, ma'am. Just give me a minute.
Yes, sure.
What about now?
Dr. Jai, you are able to see?
No, not it, not it.
Yeah, I'm not able to see either.
Yes, same here.
All right, ma'am. Is it okay if I can present after this because there seems to be some technical issue, so I can just go after the next speaker?
Okay, Joshika. Then we'll go ahead with Annie.
Ranjita will present from the group.
Ranjita will be presenting. Okay. Ranjita, you can share your screen.
Yes, ma'am.
Ranjita has background music also for us.
There's a more nearby, so.
Okay, fine. You can start sharing. I think once you start talking, we will not be able to hear background noise. Or shall we take up Malvika's, Dr. Jay? Okay, fine. I think
okay.
she started. Yeah.